Okay guys, hello and welcome to the 6th class of Science and Technology. Today we are going to finish biotechnology, most of health. Okay, so let's get started. Firstly, today we are going to read about genome editing techniques. What did I say? Genome editing techniques. What is genome? You know, it is the full complement of DNA that an individual has. And now we have read these terms, so I can go slightly faster. Genome and editing means modification. So these are the ways that we modify or edit the DNA. The terms we have read so far related to this are restriction enzymes and we have read DNA ligase, right? This we were seeing in plasmid. But plasmid mein what we were doing? In bacterial plasmid, we use restriction enzyme to cut the plasmid, generate sticky ends, then put the gene of insulin, then put that gene inside E. coli and develop human insulin. So we had used a restriction enzyme that was cutting at a palindromic site. Something like this. This site it was cutting at. Right? Now understand the problem. If I am using the same restriction enzyme in humans, this is just a 6-pair site. When I use a restriction enzyme, when it sees this site, it cuts like this. We have already discussed this. But अगर मैं humans में ये use करूंगा, तो human का तो DNA बहुत बड़ा है. Plasmid is small circular DNA. It has maybe one site like this. तो एक जगह कट लग जाएगा, दो जगह कट लग जाएगा. But human का तो बहुत बड़ा DNA है. And I want to cut at very specific place. So in a case of human, I cannot use a restriction enzyme. Because if I use restriction enzyme, it will cut at many places. So what do I need to do? I need to find a different method that can first recognize the site to be cut and then place the cut. What do we need to do? Find a different system that can one recognize where the cut has to be made. It should not be general कि जहां उसे GAA-TTC दिखा वो cut मार दे. So you need to recognize the site to be cut, and second, then you actually need to do the cut. For this, we have developed genome editing techniques. Now, genome editing techniques are as follows. There is CRISPR-Cas9. This is very important in the current affairs also, and it is the technology for which the Nobel Prize was given. So, काफी important है ये. Second is zinc finger nuclease. Third is TALEN. Fourth is site-directed nuclease. Even this is in the news. And fifth is homing endonuclease or meganuclease. So, we are going to understand all 5 of these genome editing technologies. Okay. All right. First, we are understanding CRISPR-Cas9. CRISPR-Cas9 is a way to do gene editing. It has to have a recognition. It has to recognize where I have to cut, and then it has to make an actual cut. So, 2 things are to be there. One recognition, one cut. Just a revision. What are genes? Genes are that part of DNA that code for protein. So, what will be gene editing? Gene editing will be changing the genes in humans. Okay. Gene editing will be changing the genes in humans. So for gene editing, we are using CRISPR-Cas9. Okay. Now what does CRISPR stand for? CRISPR stands for clustered regularly interspaced short palindromic repeats. Okay. Don't get scared. We will discuss this in detail. Clustered regularly interspaced short palindromic repeats. And what does Cas9 stand for? Cas9 stands for CRISPR associated protein 9. Okay. Now this system CRISPR-Cas9, I am going to explain in detail. This system CRISPR-Cas9 is basically a molecular scissor. It is a molecular scissor. It can cut something and paste something. For example, in a document, if let's say you have a Word document, what do we do? In a Word document, let's say there is some text written and you want to cut this text and replace it with some other text. So what will you do? You will press Ctrl plus F. That will be a find function. And then you will replace that text. यही करते हैं ना हम डॉक्यूमेंट में. Ctrl F, find, and then replace का ऑप्शन आता है. तो वो जो लिखा हुआ चीज है that will get replaced. Right? References 3 Idiots speech. I am sure all of you remember that. है ना? तो जो ये find और replace function है, Ctrl F से find and then replace, this reference find and replace thing we are doing at the DNA level, and this job is done by CRISPR. It is a molecular scissor. It is a find and replace mechanism at the DNA level. How does it work? Let's see. Now CRISPR was originally found in bacteria. So we will first understand how it works in bacteria and then how it has been used for humans. ठीक है. CRISPR stands for clusters of, cluster मतलब बहुत सारे, cluster of regularly interspaced. Regularly interspaced means कि बीच में बहुत सारी spaces हैं. पहले 3 लेटर I hope no doubt. Clusters मतलब बहुत सारे हैं and regularly interspaced means there are lot of spaces in between. What are these spaces? We'll see. Clusters of regularly interspaced short palindromic repeats means repeats are happening. Cluster पता चल गया, regularly interspaced पता चल गया, end में repeats are happening. And what are short palindromes? Palindrome is a DNA sequence that if I read from one side, if I read from the other side, it will look the same. ये भी हम डिस्कस कर चुके हैं. So CRISPR is nothing but clusters of regularly interspaced short palindromic repeats. Now, how does this work? Cas gene, this is palindrome interspaced by spacer DNA. Regularly interspaced है ना, तो interspaced जो चीजें हैं उसे हम कहते हैं spacer DNA. We will see all of that. The Cas gene is CRISPR associated protein 9. जो CRISPR है, the CRISPR, its main job is to do the find function. It has to find where the DNA has to be cut. The Cas gene or the CRISPR associated protein 9 has to cut. So this is the control F and this is the replace. Okay, Cas gene is the replace, CRISPR is the control F. Cas gene has 2 enzymes in it, helicase and nuclease. एंजाइम से होता है सारा काम, हेलिकेज एंड न्यूक्लियस. वर्ड ब्रेकडाउन करते हैं. हेलिकेज, डीएनए वाज अ डबल हेलिक्स, राइट? हेलिकेज विल अनवाइंड दिस लाइक दिस. ओके, हेलिकेज वो डबल हेलिक्स को अनवाइंड कराता है. एंड द अदर थिंग इज न्यूक्लियस. न्यूक्लियस क्या करता है? कट लगाता है. न्यूक्लियस क्या करता है? कट लगाता है. सो हेलिकेज एंड न्यूक्लियस, these are the 2 parts of Cas9. Clear? Okay. Now CRISPR हमें समझ आ गया, Cas9 समझ आ गया. This was originally found in bacteria. So how it works in bacteria? This is now the main question कि CRISPR Cas9 बैक्टीरिया में कैसे काम करता है. Okay? अब ये देखते हैं. This is a bacterial cell. ये जो डायग्राम है ये बैक्टीरियल सेल का है. This is a bacterial cell and ये जो चीज आ रही है ये, this is a bacteriophage. Bacteriophage means virus that infects bacteria. We are going to do viruses also in today's class, so you'll understand. Virus that infects bacteria. So what is the virus normal mechanism of infection? It will use its genetic material to get integrated with the machinery of the living cell. So bacteriophage will be a virus that uses its genetic material to integrate with the host machinery of bacteria, then use bacteria's machinery to multiply. Use bacteria's machinery to multiply. This is a normal infection. Now bacteria wants immunity. Like we have immunity, bacteria wants to protect against these viruses. So what does it do? Bacteria has certain genes. This CRISPR-Cas9 system was in bacterial genome. It was found from bacteria because bacteria was using this to protect against bacteriophages. अब देखो, मान लो यहां पर ये बैक्टीरियोफेज ने ये पर्टिकुलर डीएनए या आरएनए, या चलो लेट्स स्टिक विद डीएनए, बैक्टीरियोफेज ने अपना डीएनए इंसर्ट किया यहां पे. इट इज ट्राइंग टू गेट इनटू द genome, it is trying to integrate with the genome. अगर ये integrate हो गया, if the bacterial DNA gets integrated into the genome of the bacteria, ये वायरस ने अपना जीनोम अगर इंटीग्रेट करा लिया into the bacterial genome, what will happen? The virus will start replicating and it will ultimately kill the bacteria. So the virus now wants to prevent, the bacteria now wants to prevent the viral DNA from getting integrated. If it can prevent viral DNA from getting integrated, the bacteria will be protected. So what does the bacteria do? Okay, the bacteria will look at this viral DNA. Bacteria will look at this viral DNA. It will recognize it and it will make a simpler molecule to cut it. It will want to recognize that, okay, this is bacterial DNA and then make a molecule to cut it. So how does it do that? How does the bacteria do that? ये bacterial cell है, इसके अंदर कुछ डीएनए है, ठीक है, इसमें आ रहा था बाहर से एक बैक्टीरियोफेज या एक वायरस दैट इन्फेक्ट्स बैक्टीरिया कॉल्ड बैक्टीरियोफेज। इसके पास भी एक पर्टिकुलर डीएनए है जो ये चाह रहा है कि यहां इंटीग्रेट हो जाए। वायरस वांट्स दैट ये डीएनए, मेरा खुद का वायरल डीएनए, यहां इंटीग्रेट हो जाए और फिर मैं मल्टीप्लाई कर लूं। बैक्टीरिया वांट्स के ये काम ना हो। ओके, सो व्हाट डज द बैक्टीरिया डू? बैक्टीरिया के पास एक यहां पर सिस्टम है क्रिस्पर नाम का, क्लस्टर्स ऑफ रेगुलरली इंटरस्पेस्ड शॉर्ट पैलिंड्रोमिक रिपीट्स। सो इट हैज दिस सीक्वेंस, दिस वायरल डीएनए सीक्वेंस अपने जीनोम में भी इसने पहले से बना के रखी हुई है। जैसे ही इसको ये वायरल डीएनए दिखता है, ये अपने यहां से एक आरएनए बना देता है। इट ट्रांसक्राइब्स द आरएनए विच इज सिमिलर टू दिस। इसका जो ओरिजिनल आरएनए होना था वो वायरस अपने जीनोम से ट्रांसक्राइब करके बना देता है। फिर ये आरएनए जाके गाइड आरएनए का काम करता है। दिस आरएनए बिकम्स अ गाइड टू engulf this DNA and then it uses the Cas system to cut this DNA. I know आपको थोड़ा समझ नहीं आ रहा होगा, अभी हम डिटेल में देख रहे हैं। The bacteria has certain system like this. The bacteria has certain system like this. यहां देखो, this is viral DNA, this is bacterial DNA. बैक्टीरिया के पास ये सेम डीएनए है जो इसके पास था। क्यों है? क्योंकि पहले भी ये बैक्टीरियोफेज जिसको इन्फेक्ट कर चुका है। सो दिस इज लाइक इम्यूनिटी। ये सेम डीएनए इसके पास है। बैक्टीरिया ने क्या किया? इस डीएनए का आरएनए बनाया, इस डीएनए का आरएनए बना के इस वायरल डीएनए को जाके चिपक गया एंड देन इट मेड अ कट। यहां पे ये चीज समझा रखी है। ये क्रिस्पर सिस्टम है। वी गॉट a new viral RNA or new viral DNA. We got a new viral DNA. What did the bacteria do? It made a RNA similar to this because it had the same DNA sequence. Then it made the Cas gene. Cas was for cut, this was for find. So what will this RNA do? It will find that, क्योंकि उसके कॉम्प्लीमेंट्री है, उसका काम ही फाइंड करना है। So this CRISPR system will find the viral DNA and the Cas will then cut the viral DNA. तो viral DNA बैक्टीरिया में इंटीग्रेट नहीं हो पाएगा। This is when पहले से ये इंफेक्शन हो चुकी है। ओके, यहां लिख देते हैं दिस इज बैक्टीरियल डीएनए। ये पूरा बैक्टीरियल सेल के अंदर की बात चल रही है। बैक्टीरियल डीएनए में देयर इज दिस प्लेस वेयर इट हैज द डीएनए ऑफ वायरस 1, दिस प्लेस वेयर इट हैज द डीएनए ऑफ वायरस 2। व्हेन वायरस 1 इन्फेक्ट्स, in case virus 1 infects the bacteria what does it do? It makes RNA of this viral one जो इसके पास था, complementary to that RNA goes and finds this, then यहां से it makes a Cas goes and cuts this। इसके पास ये viral one कहां से आया? इसके पहले भी इंफेक्शन हो चुकी थी इस बैक्टीरियोफेज से, तो इसने ये याद कर लिया। Now what happens if it gets a virus that is not नोन टू इट? मान लो एक ऐसा वायरस आ जाता है जिसको इसने पहले नहीं देखा था, अ वायरस दैट इट हैज नॉट सीन बिफोर, समथिंग लाइक अ वायरस 3। लेट्स से अ वायरस 3 कम्स। नाउ वायरस 3's DNA also wants to integrate with bacteria, बट द बैक्टीरिया डज नॉट हैव इट इन इट्स सीक्वेंस, इसके पास ये नहीं है. सो व्हाट इज दिस? This is the innate response now. This was acquired response. Like in our immunity, we had memory. Bacteria also has this memory against bacteriophages. The way we were doing in our immune system, bacteria also memorizes the viruses that have infected it. And if it has survived the infection, it keeps the memory and it will kill. How will it kill? It will make a complementary RNA and then that RNA will find कि अच्छा ये मिल गया मुझे viral DNA जिसको मुझे ढूंढना था. Then the Cas will make two enzymes, one helicase will find हो चुका है, helicase जाएगा, it will unwind and nuclease will cut it. So Cas is that cutting thing and this CRISPR system is that finding thing. अब मान लो एक नया आ गया V3 which the bacteria has not seen before. Even in this case, the bacteria is able to make Cas directly. Find it, cut it and integrate it into its DNA for further infection. Although इसकी efficiency कम है, इस system की efficiency कम है. So if it gets a new virus infecting it, V3, which it does not have, the chances of the bacteria dying are more because this is the bacteria's innate immunity. But if it gets V1 or V2, which it has before, it will use the DNA of that virus, make the RNA, find it, then use the Cas to cut it. This is why this is known as a bacterial immune system. We have taken this idea from bacteria. अब इस सिस्टम को CRISPR-Cas9 क्यों कहते हैं? Cas stands for CRISPR associated protein. इसका काम तो समझ आ रहा है. इसका काम है जहां इसको इस RNA ने बता दिया वहां इसने कट मारना. So this its job is simple. The point is here, why is this system called CRISPR? This system is called CRISPR because इसका जो structure है, it is like this: spacer, repeat. Okay. So there is some spacer DNA, then a repeated palindrome, then spacer DNA, then repeated palindrome. What is this spacer DNA? This spacer DNA is that viral DNA जो इसने अपने साथ इंटीग्रेट कर रखा है. इन दिस पिक्चर एज यू कैन सी देयर इज अ ब्लू थिंग, दिस इज अ स्पेसर, देन रिपीट, स्पेसर, रिपीट, स्पेसर, रिपीट. ओके, सो व्हाट इज इट डूइंग? इट इज टेकिंग दिस, it is making the RNA that is complementary here, that's why it's called CRRNA or CRISPR RNA. It will go and attach here. Once it attaches here, it will identify, then Cas will say fine, I have to cut this, I have understood. This is the basic CRISPR-Cas9 system inside bacteria. अगर नहीं समझ आ रहा तो it's completely fine because you just need to understand that we have taken it from bacteria and bacteria uses it to guard against viruses. इतना ही relevant है हमारे लिए. The exact system, if you're not able to understand, it's fine. The other thing that I told you was that in case we get a new virus, even then it is able to cut and integrate with its genome ताकि next time it has acquired immunity, but इसकी efficiency कम है. So ये basically bacterial immunity है. This is bacterial immunity against virus. But यहाँ हो क्या रहा है? Now understand what is actually happening here. This is important. The bacteria is able to identify a DNA and able to cut it. This was our problem and this is what we needed. हमें चाहिए था कि कोई ऐसा मॉलिक्यूल मिल जाए जो बता दे यार यहाँ पे डीएनए कट करना है एग्जैक्ट. रिस्ट्रिक्शन एंडोन्यूक्लियस ना हो कि जहां उसे पैलिंड्रोम दिखा वो वहां काटने लग जाए, ये तो हमारा पूरा डीएनए ही कट जाएगा. दिस इज अ सिस्टम दैट हैज मेक्स वेरी एग्जैक्ट कट्स इन डीएनए, राइट? So if it makes very exact cuts, therefore we have used this system to then use it for human gene editing. ये spacer repeat system आपको समझ आया? It is similar to our immune system, है ना? हम भी यही कर रहे हैं, acquired and innate response. बैक्टीरिया भी ये कर रहा है. अगर उससे कोई पुराना वायरस दिखता है तो अपने में उसने उसका डीएनए स्टोर कर रखा है कि भाई आने दो, जैसे ही वो आएगा, जैसे ही वो वायरस आएगा वो उसका आरएनए बनाएगा, उससे आइडेंटिफाई करेगा, कट लगा के उसे इंटीग्रेट नहीं होने देगा. अगर उसे नया दिखेगा तो भी वो ट्राई करेगा, बट इट्स नॉट ऑलवेज सक्सेसफुल. नाउ यू माइट थिंक कि बैक्टीरिया के अंदर तो ऑलरेडी वायरल डीएनए है, तो ये तो दिक्कत हो गई, ये तो इंटीग्रेशन पहले ही हो चुकी है। नो, this part of DNA does not get transcribed and translated. Why that happens is not important for you. It only works as a defense mechanism. It does not harm the bacteria. Okay, this is how CRISPR works. Now, what did the scientists, two scientists, very brilliant scientists called Professor Jennifer Doudna and Professor Emmanuel Charpentier. Professor Jennifer Doudna and Professor Emmanuel Charpentier, they used this system to make it into our cells for gene editing. And for this, both of these brilliant professors were given a Nobel Prize. इन्होंने इस सिस्टम को यूज करके हमारे सिस्टम्स के लिए, हमारे सेल्स के लिए जीन एडिटिंग बनाई। कैसे बनाई देखते हैं। They identified this system. This is present in many bacteria, but the bacteria they use was called Streptococcus pyogenes. स्लाइड में इसका डायग्राम भी बनाया हुआ है। The bacteria that Emmanuel Charpentier and Jennifer Doudna had used was called Streptococcus pyogenes. From Streptococcus pyogenes, they took the CRISPR-Cas9 system, modified it for our use. How? Let's see. देखो, CRISPR has basically two systems. One is this CRISPR RNA. CRISPR RNA is meant to identify the virus that is coming from outside, right? So it has a CRISPR RNA. First thing, this will identify the virus that is coming, the bacteriophage that is coming. The second system it has is called the Cas9. Now Cas9 looks like this. ठीक है, एक इसमें CRISPR RNA है। देखो, the RNA's job is to identify what to cut. ये नहीं कि सब कुछ ही काट दिया, right? So CRISPR RNA identifies what to cut. The Cas9 looks like this. ये जो दो हैं, this thing, there is a helicase here. Helicase will remove from double, like the double helix structure will become single. So helicase उसको unwind करेगा, and there is a Cas9 nuclease here. The nuclease will make the cut. So it has two systems. One, the guide RNA, and the second is the nucleus that makes the cut. So what was done by these two scientists, that they modified the CRISPR to work in our cells. This is how we are using CRISPR. We have also made guide RNAs. हम क्या करते हैं, हम ह्यूमन जीनोम देख लेते हैं। जीनोम सीक्वेंसिंग एंड ह्यूमन जीनोम विल जस्ट सी। हम पूरा ह्यूमन का जीनोम देख लेते हैं। हम देखते हैं, अच्छा यार इस बंदे में यहाँ पे ये जीन खराब है। हम बंदे का पूरा डीएनए देख लेते हैं, पूरा जीनोम देख लेते हैं, कहते हैं इसमें ये पर्टिकुलर जीन खराब है। तो उस पर्टिकुलर जीन के लिए हम एक गाइड आरएनए बनाते हैं, जो हम इस CRISPR Cas9 में डाल देते हैं। तो अब क्या होगा, दिस CRISPR Cas9 विल कम, इट विल आइडेंटिफाई कि अच्छा गाइड आरएनए विल से ओके दिस इज द बैड जीन, दैट्स व्हाई दे आर बोथ शेडेड इन ब्लू. It will identify the bad gene, then it will make a cut and the bad gene will be lost. Now I can do two things. One, if the cut has been made, if a cut has been made, one, I can allow for a new gene to be introduced, or I can allow it to repair naturally. In natural repair, there is chance of mutation. So we have read that yesterday. So there are basically three options, not two, and I'll explain to you what they are. पहला ऑप्शन है, मैंने first thing I needed to do was I needed to identify where is the bad gene. This is my DNA. This is the bad gene. The CRISPR system came and it had a guide RNA molecule. The guide RNA molecule has identified this gene and the Cas9 has placed cuts. So now what we are left with is this. This is what is now left. First option is option 1. I let my cell do the recovery itself. यार खुद से ठीक कर लो. If it does the recovery itself, it will not make the bad gene. It will make the normal gene, or it will have certain mutation कि ये bad gene खराब हो जाए. Right? It will not make the exact sequence again. It will make some different sequence. At least one nucleotide will be different. At least one of ATGC will change. At this bad gene will be gone, right? Because mutation will then lead to having a non-protein expression of this bad gene. So this was the first option, that I let it repair naturally. Second option is, if I fear कि नहीं यार हो सकता है कि I cannot obviously rely that always the mutation will happen. There is a chance that the exact same gene can form again. Chance तो है ही ना इस चीज का. there is a chance that the exact same gene can form again. Ye sara tamjham will get wasted. So what do? I introduce something called a repair template. I am not introducing a foreign gene. I am not introducing a foreign gene. But to ensure that the repair does not lead to the formation of the same bad gene, I introduce a template like this. This template has been taken from some other part of my own DNA. This template has been taken from some other part of my own DNA. It is not a foreign gene. Then I ask myself, okay, now look at this repair template and make the repair. If it looks at the repair template, it will make a repair, and then the original bad gene will not be there. So this is the second option with Cas9. First is natural repair. Second option is repair template. And third option, as I'm sure you would have guessed by now, is to introduce a foreign gene. The third option is that I introduce a foreign gene with the CRISPR-Cas9. So once the cut happens, only the foreign gene will be inserted here. So I already introduced the foreign gene. This will be third option. So these are the three options I have. This is how CRISPR works. CRISPR's job is to identify. Cas' job is to cut. After the cut, 1, I can allow for natural repair. 2, I can allow for a repair template. 3, I can allow that a foreign gene is introduced. So what is this? CRISPR-Cas9 is basically a molecular cut and paste mechanism. A molecular cut and paste mechanism. And if you notice, the pasting is done differently. CRISPR's main job is to make the cut. Therefore, in every coaching compilation that you read, CRISPR is called a molecular scissor. It is called a molecular scissor. But you need to know its working so that you know its application, because UPSC has asked question on this before. And the next question will be slightly more detailed. So we have understood why it is taken from bacteria, how it works in bacteria. That part is not that important. But we have understood how Emmanuel Charpentier and Jennifer Doudna have modified it to work for humans. Now the possibilities are endless. I can cut any gene, I can paste any gene. So if I want, I can get my favorite blue eyes now. If I was doing CRISPR-Cas9 editing at my cell level, I can cut the gene that codes for black eyes and paste the gene that gets blue eyes. So this is what we have done. Here is an example working in our cells. This is the guide RNA. First part of CRISPR was guide RNA. Remember, this is what the CRISPR was doing. The guide RNA goes like this. The helicase will unwind. Guide RNA will attach here. After guide RNA has attached here, the nucleus will make the cut. So it will look like this. Virus में भी ऐसे ही कर रहा था वो. Like bacteria was doing this to the virus. Bacteria is doing this to the bacterial system is now doing this in our cells. So the first job is to open the DNA. Second job is to let the guide RNA attach. And third job is to make the cut. Now I can either let the normal repair happen, either I can introduce foreign gene, or I can introduce a repair template which is not a foreign gene. Okay. So CRISPR-Cas9 consists of single guide RNA, right? Single strand, one strand of guide RNA, CRISPR associated protein 9 for the cut, which has two things, helicase and nuclease. It is like using Microsoft Word. Control F is the single guide RNA or the sgRNA, and replace is the Cas9. Therefore, CRISPR-Cas9 is a molecular scissor. Now we have possible applications. Gene editing, of course. Gene therapy also. Gene therapy also we can do from this, right? Remember what we were doing. In gene therapy also we wanted to insert certain genes. We were using virus. Virus was going, integrating its genome. It was a difficult process. This is a much easier process. So gene editing, gene therapy, creation of genetically modified organisms. Of course, now genetic modification is so easy. I have to just cut. I know where to cut. I know where to paste. Easy job. Now the issue is this: designer babies. All cells of a baby are made from a zygote. Zygote is the first cell. It is possible that a parent wants, or when I want, that my child should have blue eyes. I might want that, okay, edit this zygote, make the zygote in the lab, edit it, put the gene for blue eyes, put the gene for tall, having a tall body, put a gene for having muscular strength, put the genes for intelligence. So I am basically toying with nature, making designer babies. I am designing what I want, and this can be done through CRISPR-Cas9. And for this, a particular Chinese scientist has been jailed as well. So this is one of the major ethical issues. One more thing I can do germline editing. So if I don't want to edit my zygote, I will just edit my germ cells. Germ cells will make sperms, sperms will make zygote, zygote will be formed into new offspring, new child. So I can edit my germ cells. If I edit my germ cells and introduce the gene for blue eyes in all my germ cells, then definitely my child will have blue eyes, right? So these are the possibilities. So now blue eyes is easy, okay? But there is some ethical problem. What is the ethical problem? That there is increased risk of mutation. Why increased risk of mutation? Because there will be a problem of cutting and allowing natural repair. In natural repair, there is risk of mutation, right? In natural repair, there is also possible that sometimes cancer genes get activated or cancer genes get formed due to this mutation, right? So this is the first risk. Second risk is designer embryos, right? Then there is impact on human variation. Humans में जो diversity है, this diversity allows us to survive many conditions like this. Remember the case of bollworms. In case of bollworms, using the idea of natural selection, what did we see? That if nature changed, if BT cotton was brought, not all bollworms died because some had the variation, some had the gene to survive even BT cotton. So the population was kept alive. But if in humans, I design and I make the genomes very similar, if everyone gets blue eyes, if everyone is tall, if everyone has muscular strength, then sometime if a disease comes that only affects tall, muscular, blue-eyed individuals, they will die, and then the entire population will die. So this variation is very important. It is very important to have very different type of genes present, or at least the variation present in the entire population. So if in nature there is some problem, selection can happen for those people who do not have that problem. If in nature there are certain conditions that are bad for particular genetic predisposition people, we want that selection can happen for those who are okay. This is the idea of natural selection given by Charles Darwin, and Charles Darwin did not say the word survival of the fittest. Okay, it is his idea, but these words were said by Herbert Spencer. Just telling out for your information. Okay, so if I use CRISPR, there will be impact on human variation. There can be abuse of genetic editing. There can be off-target effects. Here I am trying to ensure that only the targeted gene is cut. But sometimes it's possible that technology does not work. No technology is 100% proof. So sometimes it may cut DNA in places where I don't want it. What will that be called? Off-target effect. Then there is the idea of informed consent and justice and equity. Justice and equity, why? Because it's very costly, right? Since it's very costly, the rich people will have access to all of this. And informed consent, people must consent to get their DNA changed. Sometimes it's possible that doctors or genetic scientists might do this without taking the consent of the patient, without asking the patient for the permission. So this is the issues of CRISPR-Cas9. For mains also you are sorted. Now let's see where we have used CRISPR-Cas9. We have used it in precision guided sterile insects. Okay, precision guided sterile insects. Let's do word breakdown. Let's not read first. Let's do word breakdown first. Sterile. One thing is fertile. Fertile means that I have the ability to produce children. Sterile means that I don't have the ability to reproduce. Okay. Sterile insects. Precision guided, I'm sure you can understand it has something to do with CRISPR. The precision and guiding and sterile insects. So I'm making insects that don't have the ability to reproduce. Let's see in detail. Word breakdown करके हम एक basic idea लगा सकते हैं. So in prelims, if you get a term like this that you have not heard before, okay, if you get a term like this, don't be afraid. Try and find what every word means. It will not work every time, but even if it works 1 out of 3 times, you will be at net positive in prelims. Okay. Now CRISPR based system was used here to safely restrain mosquito vectors. What are mosquito vectors? Vector क्या होता है? Carrier. हमने DNA vector देखे. Mosquitoes are carrier of disease. So if an Anopheles mosquito bites you, it is a chance that you might get malaria. It is not the Anopheles mosquito that causes malaria. It is Plasmodium that causes malaria. And we will see that in today's class. But the mosquito carries the disease causing organism called pathogen. So mosquitoes are vectors of pathogens. Okay. So here what I was doing, I was using this precision guided sterile insect technique based on CRISPR to sterilize male mosquitoes. What did I do? I sterilized the male mosquitoes. How would I have done it? If in the male mosquito's DNA, there must have been some place that is responsible for producing the enzymes or proteins that are important for the production of the mosquito's sperm cells. So what I did with CRISPR, I made a guide RNA and I made a cut. A guide RNA and I made a cut. So now these enzymes will not form. So the mosquito is unable to form sperms. So the mosquito is unable to reproduce, unable to form offspring or new mosquitoes. So this is one of the way to stop mosquitoes from growing. Just stop them from reproducing. This is what was done in the case of precision guided sterile insect technology. Male mosquitoes were sterilized and female mosquitoes, female mosquitoes were rendered flightless, meaning the proteins responsible for the formation of wings at that place of the gene there was cut made. So female mosquitoes could not fly and male mosquitoes could not reproduce. It is actually the female mosquitoes in Anopheles or the Anopheles mosquito that cause malaria. It is the female mosquito that carries the Plasmodium, not the male. So female we made flightless and male we sterilized. So currently जो females हैं वो उड़ के किसी को इन्फेक्ट ना करें, मेल्स जो हैं वो आगे रिप्रोड्यूस ना कर सकें. Okay, but the problem here is that what if this gene now, this genetic editing that was happening, this kept on going on and on from generations to generations in mosquitoes. What will happen if we do this? If we do this, mosquitoes will be eliminated. But mosquitoes are part of the food chain and the food web. There must be some organisms that are eating mosquitoes, some further organisms that are eating the organisms that are eating mosquitoes. ऐसे food chain, food web होता है. I hope these basic concepts of environment are clear. If they are not, we'll discuss them, but just an idea, right? So people must be eating mosquitoes. उनका क्या होगा? Now this species will start becoming extinct, then this will become extinct, तो problem हो जाएगी. So we did not want this gene to spread. That is why it's called a precision technology. This particular technology did not get this gene transmitted of this flightless mosquito of the female. ये gene transmit नहीं होता था. This was just done for testing. This was why it was called precision guided sterile insect. इससे पहले भी एक technology has been used before. A technology called gene drive has been used before. Gene drive में क्या हो रहा था कि वेक्टर्स तो सप्रेस हो रहे थे, बट जनरेशन एक के बाद एक ये जीन पास ऑन होती जा रही थी. व्हाट इज जीन ड्राइव इज नॉट इंपॉर्टेंट, जस्ट गिविंग एन आइडिया. इट वाज पासिंग फ्रॉम वन जनरेशन टू द नेक्स्ट. सो इट वाज बिकमिंग अ प्रॉब्लम फॉर अस. So we made a PGSIT, precision guided sterile insect technique, which is self-limiting. Self-limiting meaning it does not spread. So it does not spread to all the mosquitoes in the environment. It only targets the mosquitoes we want to target. Okay, so it does not spread. How it does that is not important, but the basic idea is clear कि CRISPR यूज किया होगा, जीन को काटा होगा and stop किया होगा from the mosquito to spread or to breed. Okay, next we had started with something called genome editing techniques. Okay, we had started here. We have understood this thing. CRISPR-Cas9 is now clear to you. Okay, now there is other gene editing techniques called zinc finger nuclease, TALEN, site directed nuclease, and homing endonuclease. Now let me tell you something. The basic function of all of them is similar. The basic function is similar. Even zinc finger nuclease are cut and paste, even TALENs are cut and paste. But why is CRISPR important? CRISPR has the maximum efficiency. CRISPR has the maximum efficiency. Zinc finger nuclease and talons don't have that much efficiency. Now let's see zinc finger nuclease and talon in detail. Okay. Zinc finger nuclease are also DNA scissor or molecular scissors, much like CRISPR. Actually, this is all you need to remember. Alright. But let's see a little bit detail. The DNA binding part here is the zinc fingers. The zinc fingers are the DNA binding part, and it will recognize the portion of DNA to be cut. Why are we not using this anymore? Its efficiency is very less. Its efficiency is about 0 to 20%. This efficiency is not good enough. Okay. So DNA binding part is zinc finger, nucleus part is the cut. The nucleus that is used here is called FOC1 nucleus. Okay, and I'm sure this will be the easiest thing to remember in today's class. So it is called FOC1 nucleus. FOC1 nucleus or FOC-L nucleus. Some textbook use this term, some textbook use this term. FOC1 nucleus or FOC-L nucleus is the cutting part. What is the recognition part here? Recognition part is the zinc finger. Success rate is very low, 1 to 20%. This is an older technology than CRISPR-Cas9. Just giving a brief into how it works. Let's say this is the DNA that I want to cut. If I were to use a restriction endonuclease, what would happen? It would cut everywhere. Right? A restriction endonuclease would cut everywhere, but I want very specific recognition. So in zinc finger nucleases, what happens? Zinc fingers bind like this to certain parts. Here also they bind to certain parts. Okay. Once this binding is complete, then the FOC1 nucleus comes. The FOC1 nucleus then makes the cut in between like this. So we can identify it to bind certain parts. In restriction enzymes, what was happening? It was only recognizing 6 things. So wherever these 6 things are there, this is there. Wherever it is there, it will cut. But here, one zinc finger recognizes 3 base pairs. Means it recognizes 3 things like AUG. Let's say this zinc finger recognizes AUG. But I can combine as many zinc fingers as I want. So basically, मैं चाह क्या रहा हूं that the cut should be specific. It should be a precise cut. It should not be everywhere in the DNA. So what I do, I link many zinc fingers together. When I link many zinc fingers together, they bind at particular places and the cut it becomes very, very specific. If I link 10 zinc fingers together, then I am making a 30 sequence identification ना. जब वो 10 जिंक फिंगर्स टुगेदर होंगी तो जब 10 AUG और ऐसे करके जब ऐसे 30 चीजें आएंगी तभी वो जिंक फिंगर सारी बाइंड करेंगी और तभी कट लगेगा, राइट? सो इट कैन बी यूज्ड टू मेक अ स्पेसिफिक कट, but as you can see, linking zinc fingers at that smaller level is a very difficult process. That is why this technology was abandoned and the new CRISPR technology was gone. अगर ये नहीं समझ आया, that's fine. All you need to understand is that zinc finger nuclease, ZFN, is also a molecular scissor. The zinc is for recognition, nucleus is for cutting. The nucleus is called FOC-L nucleus or FOC1 nucleus. It has a low success rate. It is an older technology than CRISPR-Cas9. Second is talon. Talon is also a molecular scissor with the same problem of zinc finger nuclease, that the success rate is low. Other similarity with zinc finger nuclease is that the nucleus is also similar here. It is a FOC1 nucleus, but there we were using zinc to identify which part of DNA has to be recognized. Here we are using transcription activator like effector, TAIL. It is, देखो नाम देखो, TAIL N. N stands for nucleus, TAIL stands for transcription activator like effector. How this works is absolutely not important. Okay, just remember that it is transcription activator like effector. This thing, this TAIL, TALE, is the recognizer, and then FOC1 nucleus is the cut thing. Again, it has the same problem that the efficiency is low. It's not that high. Like CRISPR-Cas9 has been taken from Streptococcus pyogenes bacteria. Like CRISPR-Cas9 has been taken from Streptococcus pyogenes bacteria, talons have been taken from bacteria called Xanthomonas. It's written here. Okay, so this is also clear. Okay, this is how it works. Just एक दिखाने के लिए, इफ यू डोंट वांट टू recognize this, it's completely fine. What we do here is we take the bacterial plasmid. In the plasmid we put this talin. This talin from the plasmid codes for mRNA. The mRNA codes from tail. Talin बना देता है mRNA. So the DNA for the talin is put here. The talin then goes into the cell, codes for mRNA. mRNA codes for protein. अब प्रोटीन आ गया. प्रोटीन पहला कौन सा है? टेल प्रोटीन. टेल प्रोटीन ऐसे बाइंड करके डीएनए में आइडेंटिफिकेशन कर देता है और फिर न्यूक्लियस इसको कट करा देता है. राइट, न्यूक्लियस इसको कट करा देता है. नाउ वन डाउट आई हैड सीन इन द प्रीवियस क्लास दैट इफ आई एम पुटिंग द प्लाज्मिड, डीएनए वैक्सीन में भी तो हम यही कर रहे थे कि प्लाज्मिड हम सेल के अंदर डाल रहे थे. तो लोग कहते हैं ये प्लाज्मिड यहाँ रेप्लिकेट कैसे कर रहा है, प्लाज्मिड कैसे बना रहा है एमआरएनए, प्लाज्मिड कैसे काम कर रहा है? तो प्रोकैरियोटिक सेल का पार्ट है, हमने यूकैरियोटिक सेल के अंदर डाल दिया. When I taught you plasmid, I said something called circular, autonomous, extrachromosomal DNA. This word autonomous, why was it there? Because plasmid does not need anything else to replicate. Plasmid is autonomous. आप उसे वहां कहीं भी डाल दो, वो replication अपनी खुद कर लेता है. Right? Beyond this is not important. So in talin, what we are doing, we are putting the gene for talin in a plasmid, sending the plasmid in a cell. Talin then codes for mRNA. This DNA of talin codes for mRNA. mRNA codes for the transcription effector-like effector. Sorry, transcription activator-like effector. Sorry about that. Transcription activator-like effector protein. वो एक तरफ लगेगा, ये दूसरी तरफ लगेगा, and it will make the cut. अगर नहीं समझ आ रहा, all you need to remember, talin is also a molecular scissor like CRISPR-Cas9, but older technology. Tail for recognition, nucleus for cutting. It will make the break. One option is I will let the natural repair happen, mutated DNA, right? It will mutate and the natural repair will happen. What is this called? This is called knocking out the gene. The original gene is now knocked out because natural repair happened and mutation happened. Second is I can insert the gene, right? I can insert a foreign gene. This will be called gene knock-in because I have inserted a foreign gene. So if I let the repair happen naturally, what is happening? The bad gene is knocked out because mutation happens in that place. Second thing is I insert a foreign gene. That will be called gene knock-in. The idea of putting a repair template is also here. Repair template या natural repair में I am just letting the original gene get knocked out. Here I am getting a foreign gene, so I am knocking in a gene. Right? I hope this is clear. अब आते हैं साइट डायरेक्टेड न्यूक्लियस. दिस इज इन द न्यूज. साइट डायरेक्टेड न्यूक्लियस आर आल्सो जीनोम एडिटिंग टेक्निक. ठीक है, दिस आर द टेक्नोलॉजी हियर इज सिमिलर टू ZFN एंड टैलिन. एक्चुअली सिमिलर क्या? इट इज ZFN एंड टैलिन. ZFN and talin, zinc finger nucleus and talin have been used to create site directed nucleus. ZFN and talin have been used to create site directed nucleus. There are three types of site directed nucleus: 1, 2 and 3. Now we'll see the functions of all these three. Site directed nucleus refers to cleaving the DNA, cutting the DNA strands, and then subsequent genome editing can be done. Okay, so site directed nucleus are basically of three types. How has this technology been made? It has been made with the help of the same technology, zinc finger nucleus and talin. Okay, in site directed nucleus 1, there are three types. In site directed nucleus 1, what is the mechanism? Self repair and no foreign DNA. पढ़ चुके हैं हम ये. कट करेंगे and self repair. Second, in site directed nucleus 2, what is the technology? Repair template supplied. Repair template is supplied. Third, site directed nucleus 3. Here foreign gene is supplied. So SDN1 is self repair. SDN2 small repair template. Is this repair template a foreign gene? No. It has been gotten from some other place of my DNA. Okay. It is not a foreign gene. It is just a repair template to ensure that repair happens properly. In this case, SDN3, there is a foreign gene. So SDN1, SDN2, SDN3. These are the three technologies. अब है छोटा सा test of this question. Just one question I am asking you. Let's see आपको कितना clear हुआ है. Tell me, is SDN1 and SDN2, is this genetic modification or not. This is not genetic modification. This is genome editing. I have made the cut, but for genetic modification, a foreign gene is required, bhai. A foreign gene is required for genetic modification. Or transgenesis and genetic modification require foreign genes. So SDN1 and SDN2 is not genetic modification. It is just genome editing. Editing is happening. I am changing it, but I am not introducing a foreign gene. For genetic modification, a foreign gene is needed. Okay. Genetic modification requires foreign genes. If this foreign gene comes from a different person of the same species, it can be called GM, genetic modification. If it comes from different species, it can be called transgenesis. But I think these terms are causing confusion. So what you can do is, you can consider transgenesis and GM as the same. Itna detail mein I rarely doubt they are going to ask. So for genetic modification, remember, a foreign gene is required. The other idea is genome editing. Genome editing. Genome editing just means that whatever your genome was like, I am making some change in it. So if you had a bad gene, let's say you had a bad gene here, I am making a change in it by making a cut. Then I am letting natural repair happen. So I have changed your genome. I have edited your genome, but I have not introduced a foreign gene. Only when foreign genes are introduced, they are called genetic modification. Foreign genes, GM. No use of foreign gene is genome editing. Genome editing is broad. Genome editing का एक part है GM. Genome editing has a small part called GM, where I am introducing foreign genes. But genome editing, as we have seen here, can be done even without introducing new genes. Here also genome editing is done, but the defective gene has been changed, but no foreign DNA was introduced. So SDN1 and SDN2 are not genetic modification technology. They are, however, genome editing technology. Okay. Now, why is this in the news? SDN1 and SDN2, there is no alien genetic material that is being introduced. So the result here is not that of a GM crop. So SDN1 and SDN2, they have been exempted from the 1989 rules on GM crops because these are not GM crops. SDN3 is not exempted. SDN3, if you introduce a foreign gene in which is more than a certain length, SDN3 will come in GM rules 1989. But the 1989 rules, you will get an exemption from SDN1 and SDN2. Why are you getting an exemption? Because if you are using SDN1 and SDN2, you will never be able to tell if this is a GM crop. Because, let me give you an example. There was plant 1. There was plant 1 P1. It had certain issues, like its yield was low. Okay. Why was its yield low? Its yield was low because it had a bad gene. Okay. So if I remove this gene, its yield become normal. Now there is another way. I can make plant 1 hybridize with plant 2. Without doing any of this, I can make plant 1 hybridize with plant 2. And it is possible that their offspring gets the nice gene from plant 2 and its yield is also normal. Right? So first way is that I have plant 1. अब मैं चाहता हूँ plant 1 की तो yield low है. Now I want that the further plant it produces should not have this problem because of the bad gene. So first is I traditionally hybridize it with a second plant. The offspring might get the gene from this and have normal yield. This is first possibility, traditional hybridization. Second possibility is I make cuts and I do genetic editing. genetic editing, not genetic modification. Cut how? I do through SDN1 and SDN2. Okay, so in both these cases the result is same. It is the normal yield. So you will never be able to distinguish if I have done it through this or if I have done it through this. कोई foreign gene नहीं introduce हो रही ना, बस abnormality को ठीक कर रहे हैं. It is like a person having a gene for blue eyes. I cut that gene. Now the repair happens and now the gene is for black eyes. This is not genetic modification. I have not introduced a foreign gene. I have just cut one gene and let it repair. And in the repair process now black eyes are developed. Okay. After looking at that person, you will never be able to tell if genetic modification has happened. Genetic modification will be if I introduce the genes for making wings in him from a bird. Then he will have wings. Okay. Then that will look like genetic modification. So in SDN1 and SDN2, you cannot differentiate from traditionally hybridized varieties. And therefore, they have been exempted from the 1989 rules. Okay. Now, next thing is homing endonuclease or meganuclease. Again, this is another type of molecular scissors. That's all you need to know, right? With homing endonuclease or meganuclease, you can choose to add or not add any new genes like we had seen in other. Just similar to them, how they work is not important. काफी पुरानी technology है. Okay. Now, what is gene doping? Gene doping is when athletes change their DNA to enhance their performance. When athletes change their DNA to enhance their performance. It can be done ex vivo or in vitro. Ex vivo means outside the body. In vitro means in lab. So if certain cells are responsible for your performance, you take those cells out, modify them in the lab, give them the nicer genes for muscular function and give them back. जैसे मान लो हमारे जो joints होते हैं, उनके जो cells हैं, उनमें अगर मैं कोई gene डाल दूं बाहर से, which makes them more tolerant to running, so you will be able to run faster. I first take them out, then I inject the gene, then I put them back. This would be in vitro gene doping. Doping क्या है? Doping is using artificial methods to increase your sports performance. Artificial methods to increase your sports performance. This can be done outside the lab. It can be done in the lab. It can be done inside the body also. It can be done inside the body also, the way we were doing gene therapy through viruses. Right? So that is gene doping. Again, word breakdown से आपको clear हो जाएगा. Doping means artificial enhancer and gene तो आपको पता ही है. So using a gene as an artificial enhancer, there have been some issues with the WADA. The WADA is the body called World Anti-Doping Authority. It has issued certain guidelines on gene doping. That is why it was in the news. So this term should be clear to you. Now let's talk of animal cloning. Animal cloning and stem cell therapy. These are the next two concepts we have to discuss, and they are fairly technical, but after that there is no technical concept in today's class. Next thing is animal cloning. Okay, animal cloning. What is a clone? Clone is an exact copy. Clone means exact copy. Okay. Now, अगर humans की exact copy आपने देखनी है, तो one idea is identical twins. Identical twins are exact copies of each other, right? So identical twins, but they are coming from two parents. If there are identical twins, people who look exactly alike, they are sort of clones, but they are coming from two parents, right? One mother and one father. Animal cloning is to create a clone from one parent. That means a copy of one person. One parent, उसी का exact copy is called animal cloning. Identical twins is a natural phenomena. Animal cloning is a lab-based artificial thing that we do. Animal cloning is an artificial thing that we do. Okay, how do we do it? We do something called somatic cell nuclear transfer technique. Somatic cell nuclear transfer technique. Let's see that. Cloning is exact same genes. To have exact same genes as your parent. Exactly the same. One parent, not two. One parent uska copy. या one person uska copy, exact copy. This was first done in the case of Dolly the sheep. Dolly the sheep. This was the first ever cloned animal. क्या किया गया? We took one egg. Or let me just explain here. We took one egg cell of a normal healthy sheep. Okay. Normal healthy sheep का हमने लिया एक egg cell. अब इस egg cell में अगर normally growth होती तो क्या होता? एक egg का एक haploid nucleus होता N, एक बाहर से न्यूक्लियस आता sperm से N. These two nuclei would have fused to form first pronucleus, then the actual nucleus with 2N. तो आधा डीएनए यहाँ मदर का होता, आधा डीएनए यहाँ फादर का होता, and this would have fused to form 2N, and this 2N would then have grown from zygote to many other parts. है ना, ये zygote बनता और ये एक zygote जो एक सेल है फिर ये बॉडी के सारे सेल्स बनाता. So here the DNA was coming from two places. What happens in case of identical twins? In case of identical twins, what happens is there is a sperm, there is an egg. These fuse to form zygote. Now this zygote will form, it will have N from here, finally it will have 2N. In identical twins, twins that are exactly similar, ये zygote बनते ही दो zygote बन जाते. So this zygote splits. Naturally, it splits into two zygotes originally, and then these both independently lead to the formation of child. Normally क्या होता है या usually क्या होता है? This zygote leads to formation of one child. But कभी कभी ये zygote ही अपने आप में split होके दो बन जाता है, and then both the zygote form child. तो ये होता है identical twins में. Cloning में हम क्या चाह रहे हैं? I'll just leave this here. Here what is happening, from two parents we are forming two identical individuals, but now I want कि इसी parent का DNA आए. So what I would do here is I will take the egg has haploid nucleus N. तो egg से मैं क्या करूंगा? मैं nucleus निकाल लूंगा. I will get the nucleus out. Then I will take a somatic cell, not a germ cell, a somatic cell. Somatic cell means those cells that don't participate in meiosis and germ formation or gamete formation, they are called somatic cell. जैसे कि neck cell. तो मैंने एक somatic cell लिया, इसका न्यूक्लियस उठाया, न्यूक्लियस में सारा डीएनए है, सारी जीन्स हैं, न्यूक्लियस उठाया और यहां डाल दिया. So now this person or this egg has DNA only from one source, and somatic cells में तो 2N ही DNA होगा, नॉर्मल ही होगा. तो मैंने ये 2N DNA ही एज इट इज यहां डाल दिया. मैंने न्यूक्लियर फ्यूजन का वेट नहीं किया, मैंने प्रो न्यूक्लियस नहीं बनाया. मैंने एग को पहले उसका ओरिजिनल न्यूक्लियस निकाला, लाइक वी वर डूइंग इन प्रो न्यूक्लियर ट्रांसफर ऑल्सो. मैंने एग का ओरिजिनल न्यूक्लियस निकाला, एंड इंस्टेड ऑफ प्लेसिंग द टू न्यूक्लियाई, द टू प्रो न्यूक्लियस, आई प्लेस्ड द न्यूक्लियस फ्रॉम वन सोमैटिक सेल कैरिंग द कंप्लीट डीएनए. दिस इज सोमैटिक सेल. सोमैटिक सेल, somatic cell, what is this? Nuclear transfer. This is the name of the technology, somatic cell nuclear transfer technology. इससे होगा क्या? अब ये एग जाइगोट बन गया, और एग में जाइगोट बन गया, और इसमें डीएनए कहां से आया है? सिर्फ एक सेल से, सिर्फ एक पेरेंट से. अब ये जाइगोट आगे ग्रो करेगा तो एग्जैक्ट कॉपी बन जाएगा इसका. नाउ दिस इज नॉट दैट सिंपल, इसमें काफी टेक्निकल कॉम्प्लिकेशंस है, बट दैट इज नॉट इंपॉर्टेंट फॉर यू. वो हम दिस प्रोसेस वी हैव डन फॉर एन एनिमल कॉल्ड डॉली द शीप. डॉली द शीप की हमने मेमोरी सेल्स लिए. वी टुक मेमोरी सेल्स. मेमोरी सेल्स आर सेल्स इन द ब्रेस्ट, दे आर सोमैटिक सेल्स. उनका न्यूक्लियस निकाला, उसको डाला हमने इन द ई न्यूक्लिएटेड एग. ई न्यूक्लिएटेड एग मींस न्यूक्लियस हटाया हुआ एग, उसमें डाला. देन द जाइगोट फॉर्म्ड, फिर उससे ग्रो कराया. तो अल्टीमेटली डीएनए का सोर्स क्या हुआ? ओनली डॉली द शीप वाज द सोर्स ऑफ डीएनए. तो जो क्लोन बना दैट लुक्ड एग्जैक्टली लाइक डॉली. थोड़ा सा फर्क आएगा कि एग में माइटोकॉन्ड्रियल डीएनए भी होगा, बट लाइक आई हैड टोल्ड यू, माइटोकॉन्ड्रियल डीएनए ओनली हैज 37 आउट ऑफ द 20,000 जीन्स. So it is not a complete clone, but it is very close to a complete clone. Okay, एक बार आप ये देख भी लो. This is what we were doing. I took egg cell from one place. I took mammary cells. Mammary cells का मैंने nucleus निकाला. I put inside the egg cell. It formed a zygote. Then it grew. ये egg मैंने वापस plant कर दिया वहां उस animal से जिससे मैंने egg निकाला था. Nucleus डॉली का लेके DNA डॉली का लेके मैंने वापस डाल दिया into the mother and finally the sheep that was born it was same as dolly. तो मैंने क्या किया? I took a somatic cell. I took a somatic cell and I did its nuclear transfer into an egg. So it is called somatic cell nuclear transfer technology. Okay. Somatic cell, diploid cell with 2N, transfer into egg. From egg I have removed the haploid nucleus and transferred. अब ये क्या बन गया? Zygote with all the DNA coming from here. तो अब जब ये आगे ग्रो करेगा एक सेल सारे सेल बनाएगा तो exact clone will be formed. Okay, why is it in news? Because abhi Dolly the sheep was done by Mr. Ian Wilmot. It is written in the slide. Dolly the sheep को बनाए हुए काफी टाइम हो गया था. But China has recently cloned a wild Arctic wolf for the first time. उन्होंने recently clone किया है wild Arctic wolf को. That is why it's in the news. The technology is important. Now clone is not completely identical. पूरी तरह identical नहीं होता for two reasons. एक तो mitochondrial DNA और दूसरा it does not exactly look the same because environment का भी कुछ role होगा. Right? If I grew up in Delhi versus if I grew up in Nagaland, I would look a little bit different. Even if I was born somewhere else. Because environment also plays a role. तो बिल्कुल exactly identical नहीं होता, but it's very very similar. ठीक है? Now there are three types of artificial cloning. First is gene cloning. जब हम एक जीन की multiple copies बनाते हैं and then उस copy से product बनाते हैं. We have seen them in the case of human insulin. एक ह्यूमन इंसुलिन की जीन मैं प्लाज्मिड में डालता था, प्लाज्मिड को बहुत बार ग्रो कराता था, बैक्टीरिया बहुत बार रिप्रोड्यूस करता था, तो मैंने एक जीन को क्लोन किया. दिस इज वन टाइप ऑफ क्लोनिंग, जीन क्लोनिंग. अदर टाइप इज रिप्रोडक्टिव क्लोनिंग. रिप्रोडक्टिव क्लोनिंग इज ट्रांसफरिंग न्यूक्लियर मटेरियल आइसोलेटेड फ्रॉम सोमैटिक सेल इनटू enucleated egg. Transferring nuclear material isolated from somatic cell into enucleated egg. I think this should be pretty clear. ये हमने देख लिया. This is somatic cell nuclear transfer. And third thing is therapeutic cloning. अब रहता है कि भाई therapeutic cloning क्या है? Therapeutic cloning we will study after we have read stem cell technology. So first we should read stem cell technology. Okay. Stem cells. जब ये स्टेम सेल्स खत्म हो जाएगा, आफ्टर दैट वी विल रीड थेरेप्यूटिक क्लोनिंग. ओके, जस्ट शोइंग यू कि स्लाइड में है तो डरने की जरूरत नहीं है. ओके, नाउ व्हाट आर स्टेम सेल्स? स्टेम सेल्स, these are the cells that have the ability to divide and form many other cells, sometimes have the ability to form the entire individual. Cells that have the ability to divide and form many cells, to divide and form various things. जैसे a stem cell could divide and form a heart, a stem cell could divide and form a brain, a stem cell could divide and form a leg. These cells with this ability are called stem cells that have the ability to divide and form many different things. Right? These are stem cells. Zygote is one cell that is forming all the cells. So this is zygote is also a stem cell. Okay. Zygote is one cell that is forming all the cells. So it is also a stem cell. Now there are various types of stem cells. First type is totipotent. What does this word mean? Totipotent. Totipotent means toti से क्या समझ आ रहा है? Total. It can divide and form the total organism known as totipotent. So this is का example है zygote. Then the other is pluripotent. Pluri से समझ आ रहा है plural. Plural means it can form many organs but not all. इस एक सेल को मैं लूंगा तो मैं इसे मॉडिफाई करके कुछ भी बनवा सकता हूं, सब कुछ नहीं बनवा सकता, लेकिन आई कांट मेक इट फ्रॉम द एंटायर ऑर्गेनिज्म बट मेनी ऑर्गन्स लाइक प्लूरीपोटेंट स्टेम सेल्स। तो जब जाइगोट की सेल डिवीजन हो जाती है, जब वो थोड़ा और डिवाइड कर लेता है, राइट, पहले एक सेल होता है, देयर इज जस्ट वन जाइगोट, then it goes for many divisions and it becomes something like this. Then there are many cells, there is one cellular layer like this, one cellular layer like this, one cellular layer like this, तीन cellular layer बन जाते हैं। तो फिर ये जो फर्स्ट सेलुलर लेयर है आउटर मोस्ट, ये आपको याद करने की बिल्कुल जरूरत नहीं है, इसको हम कहते हैं एक्टोडर्म। एक्टो मीनिंग आउटर, एंडोडर्म एंड मेसोडर्म। देयर आर थ्री सेलुलर लेयर्स, once the zygote starts dividing it becomes something like this later। ओके, तीन लेयर बन जाते हैं। इस लेयर के सेल जो हैं दे फर्दर फॉर्म द नर्वस सिस्टम एंड द बॉडीज स्केलेटल सिस्टम। इस लेयर के सेल जो हैं दे फॉर्म द बॉडीज मसल, दे फॉर्म द बॉडीज इंटेस्टाइन्स एंड डाइजेस्टिव सिस्टम, और ये बीच वाले जो हैं दे फॉर्म मसल। ठीक है, तो ये तीन लेयर हैं। तो अब ये पहला जाइगोट क्या हुआ, दिस इज टोटीपोटेंट, ये तो पूरा ऑर्गेनिज्म बना सकता है। ये वाले सेल्स क्या हुए, ये प्लूरीपोटेंट हुए, ये सेल नर्वस सिस्टम में बना सकते हैं, स्किन भी बना सकते हैं, बट पूरा ऑर्गेनिज्म नहीं बना सकते, तो दीज आर टोटीपोटेंट। अब जैसे और बंदा डिवाइड हो जाएगा, when you grow and you become old and you become an adult, you will have something called multipotent. Multipotent stem cells are similar, they are similar to all, or they are contained or similar to the other cells in the organ। जैसे जब ये बंदा बड़ा हो जाएगा, when this person grows, it will get something called a bone। bone हमारी जो होती है वो अंदर से it is empty, उसके it is not completely full of calcium, उसके बीच में कैविटी होती है। इस कैविटी is where our blood cells are formed। तो यहां पर जो सेल्स हैं उनको हम कहते हैं बोन मैरो सेल्स। बोन मैरो इज सेल्स इनसाइड द बोन। बोन के अंदर कुछ सेल्स होते हैं, यहां से हमारे ब्लड सेल बनते हैं, बी सेल, टी सेल, रेड ब्लड सेल, प्लेटलेट, ये बोन मैरो के अंदर बनते हैं। तो बोन मैरो में क्या है, एक पर्टिकुलर सेल है, इसको आप स्टिमुलेट करो या बॉडी स्टिमुलेट करे तो ये रेड ब्लड सेल भी बना ले। रेड ब्लड सेल आर दी ऑक्सीजन कैरिंग सेल। इसको स्टिमुलेट करो तो ये व्हाइट ब्लड सेल भी बना ले। व्हाइट ब्लड सेल्स आर द सेल्स दैट फाइट अदर फॉरेन ऑर्गेनिजम्स, लाइक बी सेल एंड टी सेल आर व्हाइट ब्लड सेल। इसको आप स्टिमुलेट करो तो ये प्लेटलेट भी बना ले। प्लेटलेट आर दो सेल दैट लीड टू क्लॉटिंग, जो आपकी क्लॉटिंग करवाते हैं कि ब्लड ज्यादा ना गिरे। तो ये एक पर्टिकुलर सेल ये तीन बना सकता है। तो दिस इज ऑल्सो अ स्टेम सेल, बट इसकी जो एबिलिटी है वो काफी लिमिटेड है, इसे कहते हैं मल्टीपोटेंट। तो स्टेम सेल्स आर ऑफ थ्री टाइप्स, टोटीपोटेंट, प्लूरीपोटेंट, मल्टीपोटेंट। टोटीपोटेंट फुल ऑर्गेनिज्म, प्लूरीपोटेंट सम लेस, मल्टी ये दोनों होते हैं एम्ब्रियो में, और अडल्ट में होता है मल्टीपोटेंट, मतलब स्पेसिफिक टू अ पर्टिकुलर ऑर्गन, ये उसी ऑर्गन के सेल्स बनाएगा। और थर्ड है इंड्यूस्ड प्लूरीपोटेंट। इंड्यूस्ड प्लूरीपोटेंट क्या है वो हम देखते हैं। ये देखो, दीज आर स्टेम सेल्स, आप स्लाइड में इसको पढ़ लेना, दिस टेबल इज वेरी सिंपल। ये बता रहा है कि टोटीपोटेंट होता है शुरू में ऑर्गेनिज्म, फिर जब ऐसे ये तीन लेयर में डिवाइड हो जाता है देन इट बिकम्स प्लूरीपोटेंट। ओके, ऑल राइट। नाउ व्हाट आर इंड्यूस्ड प्लूरीपोटेंट स्टेम सेल्स? इंडक्शन मींस कि किसी को स्टिमुलेट किया गया है या मॉडिफाई किया गया है या बोला गया है कि आप चेंज हो। जैसे इन इकोनॉमी देयर इज इंडक्टिव प्लानिंग, राइट? तो induced pluripotent stem cells means certain cells have been modified to form stem cells। इसमें क्या करते हैं हम, we convert somatic cells to form pluripotent stem cells। somatic cells are there in adult। somatic cell का मीनिंग पता है? somatic cell are there in adult। somatic cell को modify करते हैं to form a pluripotent stem cells। इसमें क्या है, you need to modify 4 genes of a normal somatic cell। if you modify those 4 genes it will become a pluripotent stem cells। so it will be a induced pluripotent stem cell। इन 4 जीन्स को कहते हैं यामानाका जीन। and why is it called yamanaka gene? it is called yamanaka gene after the name of the scientist Professor Yamanaka who has created this technology। okay, so induced pluripotent stem cell, his name is Shinya Yamanaka and he got the Nobel Prize in 2012। उन्होंने ये 4 जीन आइडेंटिफाई करी। 4 जीन आइडेंटिफाई करके मैं क्या कर सकता हूं, नॉर्मल सोमैटिक सेल को मॉडिफाई करके प्लूरिपोटेंट स्टेम सेल बना सकता हूं। ओके, अब बताओ इसका पर्पस क्या होगा। मैंने एक सोमैटिक सेल को मॉडिफाई करा, आई मॉडिफाइड अ सोमैटिक सेल, मेड इट अ प्लूरिपोटेंट स्टेम सेल। अब इस प्लूरिपोटेंट स्टेम सेल से आई कैन क्रिएट एन ऑर्गन। इसका पर्पस है, इट हेल्प्स इन ऑर्गन ट्रांसप्लांटेशन। देखो, मान लो कल को देयर इज पर्सन ए को हो जाती है लिवर डिजीज, इनका हो गया लिवर खराब। अब इनका लिवर खराब हो गया, इनको लिवर ट्रांसप्लांट चाहिए, मतलब इन्हें एक हेल्दी लिवर चाहिए टू लिव। या किडनी ट्रांसप्लांट भी आपने सुना होगा कि किडनी दूसरे की चाहिए तभी आप जी सकते हो, आपकी किडनी खराब हो चुकी है, यू नीड अ किडनी ट्रांसप्लांट। नाउ वन ऑप्शन इज आई डू दिस ट्रांसप्लांट फ्रॉम पर्सन बी। मैं पर्सन बी के पास 2 हेल्दी किडनीज हैं, मैं इसकी एक किडनी यहां डाल दूं, ठीक है, दोनों विल बी एबल टू लिव। बट फॉर पर्सन ए, पर्सन बी's किडनी इज लाइक अ फॉरेन ऑर्गेनिज्म। पर्सन ए के लिए, पर्सन ए के टी सेल्स और बी सेल्स के लिए ये तो फॉरेन ऑर्गेनिज्म है ना, इट इज नॉट कमिंग फ्रॉम इनसाइड द बॉडी। तो क्या होता है, हमारी बॉडी ही जो ट्रांसप्लांटेड ऑर्गन है उसको अटैक करने लगती है, क्योंकि उसे लगता है ये तो फॉरेन ऑर्गेनिज्म आ गया, आई हैव टू किल दीज सेल्स। तो दिस इज द फिनोमिना ऑफ इम्यून रिजेक्शन, इम्यून रिजेक्शन ऑफ ग्राफ्ट, immune rejection of graft. Graft means this organ transplanted, this is the graft. Our immune system will reject this graft, it will say कि भाई ये graft या ये foreign ये मत लाओ organ मेरे लिए, because this is a foreign body, I have receptors to recognize foreign bodies and I will start killing it. So in this patients what we have to do, we have to give drugs that make the immune system be suppressed. हमें इन पेशेंट्स का इम्यून सिस्टम सप्रेस करना पड़ता है अगर इन्हें बाहर से किडनी चाहिए तो, बिकॉज नहीं तो खुद का ही इम्यून सिस्टम इन किडनीज को मारने लगेगा जो बाहर से आई है। सो अब हमने इनका इम्यून सिस्टम सप्रेस कर दिया। इफ आई हैव सप्रेस्ड द इम्यून सिस्टम, नाउ दिस पर्सन विल बी इजीली गेटिंग अदर डिजीजेस, इसको और डिजीज हो जाएगी कोई। इम्यून सिस्टम तो ऐसा नहीं है ना, आई कैन सप्रेस इट ओनली फॉर द किडनी, या तो मैं इम्यून सिस्टम को सप्रेस कर दूं या मैं रहने दूं। Now I have a foreign body, I don't want this foreign body, this foreign kidney to be killed, I want it to work normally, so I suppress the immune system. Now if any other infection comes, this person will not be able to deal with that infection properly. तो अब हमने क्या किया, हमने इसी बंधन पर्सन ए के किसी एक सोमैटिक सेल को लेके उसको बना दिया इंड्यूस्ड प्लूरिपोटेंट स्टेम सेल। अब इस इंड्यूस्ड प्लूरिपोटेंट स्टेम सेल को मैं मॉडिफाई करके किडनी बना देता हूं और इसी किडनी को इनके अंदर लगा देता हूं। तो बॉडी को लगेगा ही नहीं कि ये तो फॉरेन है, बिकॉज अपने से आया है ना, इट हैज कम फ्रॉम इट्स ओन सेल। सो दिस इज व्हाई इंड्यूस्ड प्लूरिपोटेंट स्टेम सेल टेक्नोलॉजी वाज गिवन अ नोबेल प्राइज, बिकॉज जो ग्राफ्ट रिजेक्शन है, जो इम्यून सिस्टम बाहर के ऑर्गन ट्रांसप्लांटेशन को रिजेक्ट कर देता है, वो इस टेक्नोलॉजी से खत्म हो जाता है। राइट, सो आई होप दिस इज क्लियर। ऑलराइट, नाउ स्टेम सेल थेरेपी का एक पर्पस तो आपने देख ही लिया, स्टेम सेल थेरेपी का एक पर्पस तो हमने देख लिया कि हमें इंड्यूस्ड प्लूरिपोटेंट स्टेम सेल बनानी है एंड वी हैव टू इंश्योर दैट इम्यून रिजेक्शन डज नॉट हैपन। व्हाट आर द अदर आइडियाज? देखो, ये एक नॉर्मल नी जॉइंट है, नॉर्मल नी जॉइंट में कुछ सेल्स होते हैं जो डिवाइड करके और सेल्स बना रहे हैं, दे आर मल्टीपोटेंट स्टेम सेल्स इनसाइड द नॉर्मल नी जॉइंट। जब अर्थराइटिस होता है, जब हम बड़े हो जाते हैं, जब डीजनरेशन हो जाती है, तो वो जॉइंट सेल कम हो जाती है। इनिशियली देयर वाज अ बैलेंस बिटवीन डैमेज एंड रिपेयर। हम चल रहे हैं, हमारी नीज को डैमेज हो रहा है, देयर इज डैमेज, एंड रिपेयर करने के लिए स्टेम सेल है, स्टेम सेल डिवाइड करके बहुत सारे सेल बना देंगे, रिपेयर करा देंगे। Then there is a joint with arthritis where the damage and repair gets imbalanced because the stem cells are low. Stem cells are low, पुराने हो गए हैं या we have gotten old, there is degeneration, अब बॉडी में उतनी ability नहीं रही, स्टेम सेल खत्म हो गए, स्टेम सेल्स are now low, so the repair is less, damage is much heavier. So when the damage is much heavier, it leads to problem of arthritis, which happens in our old folks. आपके भी घर में पेरेंट्स या ग्रैंड पेरेंट्स में आपने सुनी होगी प्रॉब्लम। तो इसका सोल्यूशन है स्टेम सेल थेरेपी। इफ आई हैव स्टेम सेल्स फ्रॉम यू, आई कैन पुट द स्टेम सेल्स थ्रू एन इंजेक्शन हेयर। मैं इंजेक्ट करता हूं यहाँ स्टेम सेल्स एंड फिर वो फिर से ग्रो करने लग जाएंगे और प्रॉब्लम सॉल्व हो जाएगी। right? I can inject the stem cells here and solve this problem. This is stem cell therapy. We are using the patient's own stem cells to provide regenerative therapy. जो डिजेनरेट हो गया था, जो स्टेम सेल्स खत्म हो गए थे, जो रिपेयर नहीं हो रहा था, जो ग्रोथ नहीं हो रही थी, उसको रीजनरेट करने के लिए मैंने पेशेंट के खुद के स्टेम सेल्स निकाले और यहां डाल दिया. तो दिस इज कॉल्ड रीजनरेटिव थेरेपी. अब यहां वेयर विल आई गेट द स्टेम सेल्स फ्रॉम? इन दिस केस बॉडी हैज सर्टेन स्टेम सेल्स. बॉडी में ऑटोमेटिकली सर्टेन स्टेम सेल्स हैं. दे आर नॉट प्लूरीपोटेंट, दे आर मल्टीपोटेंट. तो इसमें जो हिप बोन होती है, हिप बोन से मैं स्टेम सेल्स निकालता हूं और नी में डाल देता हूं. अब वो बोन स्पेसिफिक स्टेम सेल है. बोन स्पेसिफिक स्टेम सेल कैन वर्क हेयर. तो ये नी जॉइंट में बोन स्पेसिफिक स्टेम सेल्स विल वर्क. बट इन केस इफ आई वांट टू डू ऑर्गन ट्रांसप्लांटेशन, मेरे को पूरा ऑर्गन बनाना है, तो मल्टीपोटेंट स्टेम सेल से काम नहीं चलेगा, प्लूरीपोटेंट स्टेम सेल बनाना पड़ेगा. ओके. Now stem cell therapy can be used to replace dead or damaged tissues by normal functioning cells produced by stem cells. This is why it is called regenerative therapy. Now these are the names of certain diseases that can be cured with stem cell therapy. First is thalassemia, then there is leukemia. Explain कर देता हूं. Then there is osteoarthritis, osteoporosis and blindness. Blindness में तो आपको समझ आ ही रहा है. Blindness में क्या है कि आई में नॉर्मल सेल्स नहीं फॉर्म हो रहे, तो मैंने स्टेम सेल्स लिए, आई में डाल दिए, नाउ दे विल ग्रो एंड फॉर्म नॉर्मल सेल. तो ब्लाइंडनेस ठीक हो सकती है. दीज आर फ्यूचर प्रोस्पेक्ट्स, दिस इज नॉट बीइंग डन राइट नाउ. देन वी हैव ऑस्टियोआर्थराइटिस. ऑस्टियोआर्थराइटिस इज आर्थराइटिस और डीजनरेशन ऑफ बोन. उसमें कैसे यूज होता है वो हमने देख लिया. नाउ ल्यूकीमिया. ल्यूकीमिया इज व्हाइट ब्लड सेल का कैंसर. इन दैट केस जो सेल है जिसको डिवाइड करना है वो बहुत ज्यादा डिवाइड कर रहा है, तो हमने एक नॉर्मल सेल ला दिया जो नॉर्मली डिवाइड करे. दैट नॉर्मल सेल इज आल्सो अ स्टेम सेल, सो इट्स कॉल्ड ल्यूकीमिया में स्टेम सेल थेरेपी. एंड देन देयर इज थैलेसीमिया. थैलेसीमिया विल डू इन डिटेल. ओके. सो दीज आर सम डिजीजेस. अगर आपको नहीं समझ आ रहा है इन डिजीजेस का पर्पस, इट्स फाइन. यू कैन जस्ट रिमेंबर दैट इट कैन बी यूज्ड टू treat these 4 diseases, and there will be immune rejection will be minimal. If I am doing any transplant, then the immune rejection will be minimum. ठीक है. ओके. अब फ्यूचर प्रोस्पेक्ट्स क्या है कि अगर हम 3D प्रिंटिंग के साथ स्टेम सेल्स यूज करें तो वी कैन यूज स्टेम सेल्स टू फॉर्म ऑर्गन्स. बट ये हम तब देखेंगे जब हम 3D प्रिंटिंग पढ़ लेंगे. ठीक है. Why were they in news? Because recently the Indian Council of Medical Research, it has released certain guidelines for stem cells. गाइडलाइन रिलीज हुई है स्टेम सेल्स के लिए. There is another term called stem cell banking. Now ये important है. Stem cell banking क्या होती है? When a baby is born, okay, when a baby is born, it has an umbilical cord. You have a belly button. Belly button is connected to the mother. Okay, mother की body से connected होता है belly button, right? When a baby is born, baby का umbilical cord belly button से एक cord connect होती है to the mother. जब delivery होती है, we cut this cord. And the baby can then grow on its own, right? इस umbilical cord में, umbilicus means this belly button. Belly button में we have this hole here because there is a cord there originally. इस umbilical cord में there are stem cells of the baby present. तो हम क्या करते हैं, जब हम कट करते हैं इस कॉर्ड को, जब बेबी की डिलीवरी हो गई, अब बेबी की डिलीवरी हो गई, मैंने ये कॉर्ड कट कर दी. जब मैंने कॉर्ड कट कर दी तो इसके अंदर बेबी के स्टेम सेल्स थे. I took these stem cells and I started storing them at ultra cold temperature ताकि वो alive रहें. मान लो बेबी को बाद में कोई ऑर्गन चाहिए होगा तो मुझे इंड्यूस प्लूरिपोटेंट स्टेम सेल बनाने की झंझट नहीं करनी पड़ेगी. I will just be able to take these stem cells and give this baby therapy. This is called stem cell banking. This is storage of umbilical cord stem cells at 170 degree with liquid nitrogen to be used for future therapy. Okay, stem cell banking and induced pluripotent stem cells. I hope this is clear. Now what are the challenges? Challenges तो एक तो यार we are doing synthetic biology. We are interfering with nature. There are biosafety concerns. This technology can obviously be abused. There are of course ethical concerns with regard to हम stem cells को use कर सकते हैं या नहीं कर सकते, and are we not killing people और killing embryos to get stem cells, वो कैसे होता है वो अभी बताता हूँ. And then there is genetic divide. Genetic divide, again, you know what this means. Some people are rich, some are poor. अब इन ethical concerns के बारे में आपको तब समझ आएगा जब हम therapeutic cloning पढ़ लेंगे. Therapeutic cloning is what? Therapeutic cloning वो है जो मैं आपको बता रहा था जिसके लिए हम stem cell banking करते हैं. मान लो मेरा liver खराब हो गया. Now I have stem cells that are banked somewhere, right? I want those stem cells to grow a healthy liver and put that healthy liver inside me so that there is no immune rejection. So my body does not feel that these are foreign livers. Body does not reject that क्योंकि वो stem cells मेरे खुद के हैं. So there will be no foreign immune rejection. कैसे बनेंगे? How will I use the stem cell to form the liver? I will take the stem cell. Okay, I will take my body stem cell and I will make it grow into a small embryo. मैं इससे एक small embryo बनाऊंगा. From this embryo I will take liver and put it inside me. I will, right? अब ये मेरा ही stem cell था. This was my banked stem cell. When I was born, my umbilical cord was cut. From here I got this stem cell. When I need a liver, I am growing the stem cell into an embryo. Then this embryo will grow. जब ये थोड़ा बड़ा हो जाएगा, इसका भी खुद का एक liver होगा. I will take its liver and plant it here. Right? Exact technology is not important. But then this embryo will die. क्योंकि इसका तो मैंने liver निकाल लिया ना. So is it not killing of this new life that I have formed? Can this be called life? Is this not life? ये ethical dilemma है इसमें. This is the ethical problem, and this process is therapeutic cloning. Okay, we are producing a healthy replica of a human organ, better for transplanting organs. मैंने खुद के stem cell से एक replica बनाया, right? And पूरा replica नहीं है, it's not a complete human, but it is still alive. Liver is also living, the cells are living. अब वो life है या नहीं है? This is the ethical divide here. Okay. It is like reproductive cloning. It is like reproductive cloning. It is like somatic cell nuclear transfer therapy. सेम ही बात हो रही है. मैं अपना ही sort of clone बना रहा हूँ through a stem cell. But इसमें बात यहाँ पे है कि जो embryo बनता है, it is made to grow in a lab. And then इस embryo से मैं वो liver वाले cells निकाल लूंगा. वो cells जो originally divide करके liver बनाते थे, I will extract those stem cells that are forming a liver and then put them in my body. This is therapeutic cloning. It has certain ethical issues called divine command theory or Christian ethics. Actually, Christian ethics say that even if there is one alive cell, it is just to be considered alive. They say life begins at conception. So Christians believe, or Christian ethics say, that this would be the murder of a living child. And this is, you can agree with it or disagree with it, but this is an ethical dilemma in therapeutic cloning. Okay. So now we have come to the end of biotech का सारा कुछ. Health में we have done vaccines. Let's quickly see the applications of biotech. This is just for your mains. It is not for your prelims. तो अगर आपको ये सब कुछ याद नहीं भी रहता तो भी ठीक है. I'll just run through this quickly. We'll take a 5-minute break and then do health. So application of biotech अगर आपको कभी लिखना हो, you use certain colors which is used to divide biotechnology. So there is green biotechnology in agriculture. Now green biotech can obviously be used for increased yield, to create biopesticides. Biopesticide means those plants that don't require pesticide. जैसे कि BT brinjal. Pesticide चाहिए ही नहीं. It has Bacillus thuringiensis का gene. It already protects against bollworm, so it is a biological pesticide. Then there is high yielding variety, जैसे कि GM mustard. Then there is biofortification, जैसे golden rice. ये हम सब देख चुके हैं. Then there is shelf life. What does shelf life mean? मैं प्लांट की जीन्स एडिट करके ऐसा बना दूं कि शेल्फ पे जब वो पड़ा है वो रॉट ना हो. इट डज नॉट गेट रॉटन. इट कैन स्टे ऑन द शेल्फ फॉर अ वेरी लॉन्ग टाइम. इट कैन स्टे स्टोर्ड फॉर अ वेरी लॉन्ग टाइम. तो इसमें क्या होता है, एक्सपोर्ट में हेल्प होगी. जब मेरे को एक्सपोर्ट करना होगा तो प्लांट यहाँ पे होगा. मैंने टमेटो को यहाँ से निकाल लिया. नाउ आई हैव टू एक्सपोर्ट दिस टमेटो टू यूरोप. सो इट विल टेक 30-40 days फॉर दिस टमेटो टू गेट टू यूरोप. Right? So it cannot get rotten in those 30-40 days. This can be done through green biotechnology. Then we can fight diseases. Of course, we can do RNA interference. This we have seen. Then we can develop better breeds. So this is green biotechnology. डोंट कंफ्यूज दिस विद जेनेटिक एडिटिंग और जीएम. बायोटेक्नोलॉजी का एक पार्ट है वो. इवन ट्रेडिशनल हाइब्रिडाइजेशन इज पार्ट ऑफ बायोटेक. ओके, ग्रीन बायोटेक में एग्रीकल्चर है. These are some of the examples that you can take directly to your mains. Then there is blue biotechnology. Blue biotechnology is for marine organisms that are in the sea. जैसे कि aquaculture of seafood, meaning growing fishes in the ponds. Transgenic fish. Now this is genetic modification. I put a gene inside these fish. So when I have to export them, when I have to transport them, they don't get frozen. Or when I am taking them from cold parts of the world to warm parts, or warm parts to cold parts, they don't get frozen. So this is called freeze-resistant fish. कैसे काम करता है important नहीं है, just get the idea. I can use it for conservation of certain species because I will have their genes. तो मैं उनसे conserve करा सकता हूँ, उनको clone करा सकता हूँ. And I can do cultivation of seaweeds like agar. These seaweeds are very good for your body. This is blue marine biotechnology. Then there is red biotechnology. Red biotechnology is in health. Health में तीन चीजें होती हैं: prevention, diagnosis, treatment. What is prevention? Prevention में I can first make transgenic plants. Transgenic plants ऐसे बनाओ जिससे आपको nutrition better मिले. You will get better food, right? So when you get better nutrition and food, you will not have disease. प्रिवेंट हो गई ना डिजीज, आपको बेटर न्यूट्रिशन मिल रहा है थ्रू ट्रांसजेनिक प्लांट. एप्लीकेशन 1. सेकंड इज ट्रांसजेनिक एनिमल बना दूं टू स्टडी ह्यूमंस एंड ह्यूमन डिजीजेस. ये भी हम देख चुके हैं. तो दिस इज आल्सो प्रिवेंशन. आई एम स्टडिंग ह्यूमन डिजीज बिफोर हैंड, तो उसे कैसे प्रिवेंट करना है मुझे पता चल जाएगा. एंड थर्ड इज वैक्सीन. वैक्सीन तो हम बना ही सकते हैं, वैक्सीन आर प्रिवेंशन. देन फॉर डायग्नोसिस वी कैन डू पॉलीमरेज चेन रिएक्शन, पीसीआर, जैसे हम कर रहे थे आरटी-पीसीआर टेक्नोलॉजी. We can make DNA probes. What these are is not that important, but DNA probes are meant to see if your genome has any foreign DNA. तो अगर आप में कोई वायरस आ गया तो DNA probe बनाया जा सकता है to see if your genome has any foreign pathogenic DNA. Then there is monoclonal antibody, MAB. Monoclonal antibody से आपको समझ आ ही रहा होगा कि these are antibodies. These antibodies are used in diagnosis of disease, ये हम कल देखेंगे. And there is ELISA. ELISA is also a diagnostic test. Enzyme linked immunosorbent assay, ये दोनों चीजें हम कल देखेंगे. Then there is treatment. Treatment में I can make human insulin, I can do gene therapy, I can do stem cell therapy, I can make recombinant proteins to treat you. So this is diagnosis, prevention and treatment of red biotechnology. Then there is environmental biotechnology. Environmental biotechnology, using biotechnology for environmental purpose. इसमें एक चीज है ऑयल जैपर बैक्टीरिया. दिस वाज मेड बाय टेरी. ये ऐसा बैक्टीरिया था जिसमें ऐसी जीन्स थी कि अगर ऑयल स्पिल जहां हो जाता है वहां इस बैक्टीरिया को छोड़ दो तो ये वो सारा ऑयल अब्सॉर्ब कर लेता था. तो दिस वाज मेड ऑफ कोर्स थ्रू जेनेटिक मॉडिफिकेशन. तो दिस इज ऑयल जैपर बैक्टीरिया यूज्ड इन बायो रिमीडिएशन. बायो रिमीडिएशन मींस रेमेडिंग और सॉल्विंग द बायोलॉजिकल प्रॉब्लम. तो बायो रिमीडिएशन के लिए वी यूज्ड ऑयल जैपर बैक्टीरिया डेवलप्ड बाय टेरी इन इंडिया कॉल्ड ओलियोवोरस एस. ये बैक्टीरिया का नाम है. सो दिस इज यूजिंग बायोटेक्नोलॉजी फॉर एनवायरमेंटल पर्पस. देन देयर इज फाइटोरिमीडिएशन. फाइटोरिमीडिएशन इज बायोरिमीडिएशन बट डन यूजिंग प्लांट्स. प्लांट से आप कर रहे हो बायोरिमीडिएशन, एनवायरमेंट को ठीक कर रहे हो. यहां पे वी डू यूजिंग यूकलिप्टस. यूकलिप्टस को प्लांट कर दो, वो पॉल्यूशन को अब्सॉर्ब कर लेता है फ्रॉम द सॉइल, राइट. इफ वी एडिट इट, वो टॉक्सिक एलिमेंट्स को अब्सॉर्ब कर लेता है. दिस इज फाइटोरिमीडिएशन. थर्ड एप्लीकेशन इज डीएनए बार कोडिंग. व्हाट इज डीएनए बार कोडिंग? स्पीशीज का डीएनए लिया, पढ़ चुके हैं कई बार, उसको बार कोड बना दिया. कहां मिलेगा स्पीशीज का डीएनए और यूज क्यों करते हैं? मान लो आप वाइल्ड में हो, यू आर एन इंडियन फॉरेस्ट सर्विस ऑफिसर, ठीक है. Now you are in the wild and you get some part of a particular animal. आपको एक एनिमल का हेयर पड़ा हुआ मिल गया. Now you don't know ये कौन से एनिमल का है. तो आपने क्या किया, इसका डीएनए एक्सट्रैक्ट किया. हेयर में भी डीएनए होता है, हर जगह डीएनए होता है, एवरी सेल हैज डीएनए. तो आपने क्या किया, इसका एक्सट्रैक्ट किया डीएनए. आपने उस फॉरेस्ट में जितने भी प्लांट्स रहते थे, जितने भी एनिमल्स रहते थे, उन सब का डीएनए बारकोड बना रखा है. When you extract its DNA and you match it you will be able to identify कि अच्छा ये कौन सा organism है. So you can use it for identification, but you will not be able to tell age because DNA does not tell with age, does not change with age. मेरा DNA वही है जो मेरे zygote का था. So when I am 5 years old, my DNA will be same. When I am 30 years old, my DNA will be same. So you will not be able to use DNA barcoding to assess the age, but you can use it for identification. Now we have forensic biotechnology. Forensic biotechnology is basically DNA fingerprinting, using DNA to identify ये DNA किसका है. यहाँ we use a technology called VNTR, variable number of tandem repeats. We'll just see that. But इसका use कहाँ कहाँ हो सकता है पहले वो देखते हैं. Criminal investigation में definitely it can be used. Paternity testing. Paternity testing means there is a baby. ये बच्चा actually किसका है? If it is from a particular person, its DNA will be similar, like we have seen. So if two people are claiming to be the father of one baby, we can check the DNA of the baby, DNA of person 1 and DNA of person 2. जिससे ज्यादा similar होगा उसी का बच्चा होगा ये, right? So this is using it DNA fingerprinting for paternity testing. DNA fingerprinting is basically identification of DNA. जैसे fingerprints are identifier, you are using the DNA as an identifier. कैसे करते हैं वो भी देखेंगे. In cases of child swapping, जैसे you are in the hospital, your child is born. बहुत सारे बच्चे एक दिन born होते हैं. All of them are put inside the nursery. अब nursery में usually tagging is done that this is whose baby etc. But let's say in the nursery some swapping happens. तो आप DNA compare करके बता सकते हो कि ये इन parents का है और ये इस parent का है, right? Now how is DNA barcoding or DNA fingerprinting done? This is your DNA. 2 स्ट्रैंड नहीं बनाया, सिंगल स्ट्रैंड बनाया. इसमें कुछ नॉन कोडिंग रीजन है, राइट? ये हमें पता है. नाउ इट इज इन दीज नॉन कोडिंग रीजन दैट देयर इज गोइंग टू बी द डिफरेंस. ये भी हमें पता है कि जो मेन डिफरेंसेस हैं दे आर गोइंग टू बी इन द नॉन कोडिंग रीजन, नॉट इन द कोडिंग रीजन. तो इन नॉन कोडिंग रीजन में ऐसे करके कुछ रिपीट्स होते हैं. जैसे यहाँ पे AGAC, AGAC, AGAC is repeating over and over again. This repeat is called variable number of tandem repeats. Since it is the non-coding region, it does not have to be similar. So the repeat, अब देखो variable number of tandem repeats. Repeat का तो मतलब समझ आ गया कि ऐसे एक sequence बार बार repeat हो रही है. Variable number means सब में ये number different है. Everybody has a different number of these repeats. Tandem means repeat एक के बाद एक हो रहा है. So this is called VNTR, variable number of tandem repeats. जो हमारा DNA होता है in all humans 99% of the DNA is the same. In all humans 99.9% of the DNA is the same because all humans look very very similar. The only difference is in this 0.1% DNA. This 0.1% DNA is mostly in the non-coding region obviously because coding तो बहुत similar है. So in the non-coding region there is 0.1% difference. This DNA is called satellite DNA. The similar DNA is called bulk DNA. So satellite DNA है non-coding. Satellite DNA है non-coding region में that part that is different. The 0.1% is the satellite DNA. इस सैटेलाइट डीएनए से हम देखते हैं कि दोनों में क्या डिफरेंस है. द टेक्नोलॉजी वी यूज इज वेरिएबल नंबर ऑफ टैंडम रिपीट्स बिकॉज दिस सीक्वेंस ऑफ रिपीट्स किसी में मान लो AGAC 4 टाइम्स रिपीट हो रहा है, ठीक है, और किसी में AGAC 10 टाइम्स रिपीट हो रहा है. तो ऐसे बहुत सारी सीक्वेंसेस हैं. दैट इज हाउ वी डू इट. नाउ अ स्मॉल डेमोन्स्ट्रेशन ऑफ हाउ दिस टेक्नोलॉजी वर्क्स. वी विल फर्स्ट टेक द डीएनए, cut it using restriction enzymes into many small parts. ठीक है, फिर हम यहां यूज करते हैं जेल इलेक्ट्रोफोरेसिस. तो जिसके रिपीट्स ज्यादा हैं their DNA will be bigger, जिसके रिपीट्स कम हैं उनके DNA will be smaller, एंड साइज के बेसिस पे फ्रैगमेंट्स देख के वी सी ऐसा एक पैटर्न बन जाता है एंड वी यूज दिस पैटर्न टू कंपेयर. हाउ दिस वर्क्स इज नॉट इंपॉर्टेंट, इसलिए मैं डिटेल में नहीं जा रहा. इफ यू वांट टू सी इट एवरीथिंग इज रिटन इन द स्लाइड. द थिंग्स यू नीड टू रिमेंबर आर variable number of tandem repeats. This is the technology used in DNA fingerprint. अब हम थोड़ा सा फास्ट चलेंगे एटलीस्ट इन चीजों में जो आपको आती है. जीनोम सीक्वेंसिंग. आई एम नॉट एक्सप्लेनिंग व्हाट इट मीन्स, आई होप यू नो व्हाट इट मीन्स. कंप्लीट कॉम्प्लीमेंट ऑफ डीएनए, उसको सीक्वेंस करना है. अभी रिसेंटली क्या हुआ है, एक इंटरनेशनल टीम ने पूरा genome, human genome sequence कर दिया. Like the entire genome was sequenced. Earlier we had something called a human genome project. This finished in 2003, but it did not read 8% of the genome. It was only able to do 92% of the genome. So human genome project में only 92% of the genome we could sequence. But now some team has done the rest 8% also. Human Genome Project was started in 1990. What they did, they tried to find all the base pairs, all this AGCT of the human genome from one end to the other end, the complete genome. What they found out that in human genome there are 3 billion base pairs. Base pair means जैसे A के सामने T आता है, right? This is one base pair, A and T. This is one base pair. Pair हो गया ना, A के सामने T आता है, G के सामने C आता है. So there were 3 billion base pairs. 3 billion base pairs in human genome when they sequenced it. Then they found that only 3% of the DNA codes for protein. 97% of the DNA is non-coding junk DNA. So only 3% is genes, 97% is junk DNA. There was two things, HGP read and HGP write. HGP read, Human Genome Project to read. Read was the project which wanted to sequence the genome. Here India did not participate. The idea was to build a standard human resource consortium genome. मतलब एक standard human genome build कर लेते हैं हम. Let's see what are the genes or the genome, what are the sequence of the genome of a standard human. Then we can use it. Then we can identify the genes. Then we can see how viruses act on it. Then we can see how drugs act on it. So this genome project wanted to see the entire genome, the entire sequence, but it was unable to do that. It was only able to see 92%, not the complete genome. So Human Genome Project was able to see nearly 92%. The 8% was left, right? Now what were the uses of this genome sequencing? ये important है. Genome sequencing करके हमें पता क्या चला? First, we could do disease diagnosis. हम आइडेंटिफाई कर सकते थे वो जीन्स जो ज्यादा डिजीज कॉज करती हैं, जैसे कि कैंसर की जीन, कुछ रेयर डिजीज, कुछ न्यूरो डिजेनरेटिव डिजीज, इनको हम देख सकते थे कि यार ये कौन सी जीन है एंड व्हाट आर दे कॉजिंग. देन वी कुड सी वायरल डिजीजेस एंड देयर इंपैक्ट, बिकॉज नाउ वी हैव अ रेफरेंस जीनोम. व्हेन वायरस कम्स एंड इंटीग्रेट्स its DNA into our genome, we will see the difference. So we can do viral disease and their impact study. Then we can study genetic variation. We can study the variation in the genome. We have a reference genome, so I can see the variation from one person to the next person. Then racism as an idea was completely debunked. This is very important for anthropology, not that important for you, but it was found that the differences between human DNA or differences between the whole complement, differences between the human genome are so less that humans cannot be effectively classified into races. Okay, नहीं समझ आया तो कोई बात नहीं, जस्ट समझ लो कि इतना डीएनए सिमिलर था कि हम कह ही नहीं सकते कि ह्यूमंस की अलग अलग रेसेस हैं. तो व्हाट अबाउट रेसिज्म इज नॉट अ, लाइक इट्स नॉट पॉसिबल दैट वन रेस इज बेटर देन द अदर, क्योंकि ह्यूमंस हैव नो रियल रेसेस. ओके, दिस इज नॉट इंपॉर्टेंट, जस्ट फॉर योर इंफॉर्मेशन. देन अब हमारे पास एक रेफरेंस जीनोम है. अब मेरे पास एक रेफरेंस जीनोम है तो मैं जीन थेरेपी भी कर सकता हूं. मुझे पता चल गया ये जीन यहाँ पे खराब होती है, यहाँ पे कट लगाऊंगा, यहाँ से जीन इंसर्ट करूंगा, राइट? एंड आई ऑल्सो लर्नड अबाउट एवोल्यूशन. आई लर्नड अबाउट द एंटायर ह्यूमन डीएनए. सो आई कंपेयर्ड इट नाउ विद द चिंपैंजीज डीएनए. आई कंपेयर्ड इट नाउ विद द डॉल्फिन्स डीएनए. So I found that our DNA was much closer to chimpanzees DNA. So what does that mean? It means that we have evolved from chimpanzees. तो evolutionary studies में भी इसका help हुआ. Okay. अब क्या हुआ? The 8% HGP could not do. 8% HGP could not do. It was found कि यार ये ना heterochromatin है. I'll tell you what that means in a minute. It was found that this 8% does not code for that many proteins, or maybe does not code for any protein, and it is very difficult to sequence. So the HGP team said कि छोड़ दो, let's leave this 8%, let's focus on the important 92%. This is what I will tell you also. There is an 80-20 rule in prelims. Okay, 80% of the questions will come from 20% of the material. Focus on that. Leave the rest aside. पहले वो कर लो, get that sorted, and then you chase the remaining 20% of the questions. So that's what HGP also did. इन्होंने कहा छोड़ दो 8% को, but कुछ पागल तो होते हैं. So there was a team like telomere to telomere project जिन्होंने कहा हम ये 8% भी सीक्वेंस करेंगे. So this 8% was done by telomere to telomere project, and they found that even here there are certain genetic variabilities. Even here there are certain genetic diseases that are important. So this sequencing was done by telomere to telomere project. Now again, what is the significance of genome sequencing? Genetic diseases, evolutionary biology, then how our genome works, how virus work on genome, how drugs or pharmaceutical medication when given to you, how it affects the genes. ये सारी चीजें हम समझ पाए यूजिंग ह्यूमन जीनोम प्रोजेक्ट. नाउ टीलोमेयर टू टीलोमेयर प्रोजेक्ट की बात करते हैं. जो क्रोमोसोम होता है, क्रोमोसोम इज नथिंग बट कंडेंस्ड डीएनए. क्रोमोसोम हैज टू पार्ट्स. ये वाला पार्ट आप देख रहे हो, दिस इज डेंस पार्ट, राइट. इट इज स्टेनिंग डार्कर. और ये वाला पार्ट देखो, इट इज नॉट स्टेनिंग दैट मच, राइट. तो ये जो इसे कहते हैं हम यूक्रोमैटिन. यूक्रोमैटिन, वी हैव सीन व्हाट यूक्रोमैटिन मींस. We have seen what chromatin means. Chromosome forms from chromatin. There is DNA. DNA forms to chromatin. Chromatin forms to chromosome. यही हमने पढ़ा था फर्स्ट क्लास में. तो यहां पे भी वही चीज है. क्रोमैटिन के दो पार्ट हैं. There is euchromatin and heterochromatin. Euchromatin is this light part that codes for protein, which is easier to sequence. So the HGP team, Human Genome Project, saw this and they coded all the euchromatin. उन्होंने euchromatin की सीक्वेंस निकाल ली, बट जो heterochromatin था, they let that aside. Heterochromatin is more condensed, it stains darker, and it is gene poor. It does not have many genes. But this heterochromatin also, this 8% also has now been sequenced by a team called telomere to telomere project. And why is the name telomere to telomere? ये भी रटने की जरूरत नहीं है. क्रोमोसोम का जो ये वाला पार्ट होता है, this is called telomere. So the name of the project was only telomere to telomere. So we will sequence the entire, बीच का हम कुछ नहीं छोड़ेंगे, right? So they have sequenced even the remaining 8% part. इसमें important क्या है? इसमें important है applications of genome sequencing. Think wide when you are thinking these applications. काफी मैंने बता दिए, but अपना दिमाग भी आपने लगाना है. Now you know what genes are, so think wide. Okay? All right. Now, what are some Indian initiatives for genome sequencing? Indian initiatives are two. First is the IndiGen project. This IndiGen project is run by CSIR. CSIR is Council for Scientific and Industrial Research. CSIR is running this IndiGen project. They are doing the whole genome sequencing of 1000 individuals of Indian origin. Why? जब HGP किया गया, Human Genome Project किया गया, it was done on a European population. Remember? Global North, those countries have all the money. So जो reference genome बना है, that reference genome is of a European person. But obviously कुछ difference तो होगा, right? So we want to see what is the reference genome of an Indian person, ताकि हम अपनी research और better कर सकें. अभी सारे drugs, सारे pharmaceuticals की research उस genome पे हो रही है, but maybe it's not suitable for our bodies and our environment. So that's why CSIR has launched IndiGen project. The Department of Biotechnology has also launched Genome India project. Genome India project क्या है? They will take 1000 genetic samples from citizens across India to build a reference genome. Similar ideas हैं. Just remember what genome sequencing is. These are the project IndiGen by CSIR and Genome India by Department of Biotechnology. Okay. अब viruses, which I had promised we'll do. इसमें एक चीज मैं आपको बता देता हूं. I will teach you some things now. इसके बियॉन्ड इंपॉर्टेंट नहीं है. अगर डाउट्स रहते हैं वी विल डू दैट एट द एंड ऑफ द क्लास. जिसको प्रीलिम्स पे फोकस करना है, जिसके और सब्जेक्ट रहते हैं, जस्ट ये बेसिक पढ़ेंगे वो और आगे चलेंगे. जिसको साइंटिस्ट बनना है वो रुकेंगे मेरे साथ आफ्टर 2:30. वायरसेस क्या है? दे आर बॉर्डर लाइन बिटवीन लिविंग एंड नॉन लिविंग. ये बात हमें पता है. दे आर नॉन सेलुलर ऑर्गेनिज्म. इनका कोई सेलुलर स्ट्रक्चर नहीं है. ये क्या है? दे बेसिकली दे आर जस्ट जेनेटिक मटीरियल एंड प्रोटीन. Viruses are only genetic material and protein. They have a genetic material. This genetic material could be RNA, could be DNA, and they have proteins. These proteins are of various types. Viruses में जेनेटिक मटेरियल है, प्रोटीन है. जेनेटिक मटेरियल कुड बी आरएनए और डीएनए. प्रोटीन आर ऑफ वेरियस टाइप्स. कौन से टाइप्स विल सी. जो प्लांट को इन्फेक्ट करते हैं वायरसेस, जनरली वायरसेस दैट आर इन्फेक्टेड प्लांट्स, दे हैव सिंगल स्ट्रैंड आरएनए. मीनिंग व्हाट, वायरस ऐसा है, ठीक है. दिस इज नॉट द राइट वे टू ड्रॉ इट. दिस इज अ सेलुलर स्ट्रक्चर. एस मतलब ओके, आई विल ड्रॉ द एग्जैक्ट थिंग ओनली. वायरस इज लाइक दिस, ठीक है. इट, ये क्या है, ये जो रेड में बनाया है, दीज आर ऑल प्रोटीन्स. इट हैज सर्टेन जेनेटिक मटेरियल अंदर, यहां पे इट हैज जेनेटिक मटेरियल. अब कई वायरसेस में तो बस एक स्ट्रैंड का जेनेटिक मटेरियल है, कई वायरसेस में दो स्ट्रैंड है. तो कई वायरसेस विल लुक लाइक दिस फ्रॉम द इनसाइड एंड मेनी विल लुक लाइक दिस फ्रॉम द इनसाइड. ओके, सम वायरसेस हैव आरएनए, some viruses have DNA. Okay, so what are the possible combinations? There are 4 possible combinations. A virus can have single strand RNA, virus can have double strand RNA, virus can have single strand DNA, virus can have double strand DNA. This is the possibility. Please don't go into ये वायरस काम कैसे करता है, इसके प्रोटीन्स क्या हैं, एक्सेट्रा. दैट्स नॉट इंपॉर्टेंट फॉर यूपीएससी. अगर करना है तो करेंगे व्हेन यू वांट टू बिकम अ वायरोलॉजिस्ट, आराम से कभी बैठ के. ओके, सिंगल स्ट्रैंड आरएनए हो सकता है, डबल स्ट्रैंड आरएनए हो सकता है, सिंगल स्ट्रैंड डीएनए एंड डीएसडीएनए. दीज आर द 4 ऑप्शंस. बिकॉज उसके पास पक्का कोई ना कोई न्यूक्लियर मटेरियल होगा. कभी भी डीएनए और आरएनए बोथ नहीं होंगे, ओनली वन विल बी देयर. सो इट विल ईदर बी डीएनए और इट विल बी आरएनए, राइट. एंड दीज आर द पॉसिबल कॉम्बिनेशंस. इसके अलावा उसमें क्या होंगे, इसमें होंगे प्रोटीन्स. तो अब जो हम क्लासिफिकेशन देखते हैं वायरसेस की, उसमें क्या पता चलता है कि जो प्लांट को इन्फेक्ट करने वाले वायरसेस हैं, दे जनरली हैव सिंगल स्ट्रैंडेड आरएनए. जो एनिमल्स को इन्फेक्ट करने वाले वायरसेस हैं, दे हैव ईदर सिंगल और डबल स्ट्रैंडेड आरएनए और डबल स्ट्रैंडेड डीएनए. ओके, दिस इज रिटन इन द स्लाइड. सिंपल लाइन है, रीड कर लेना. आई हैव एक्सप्लेन व्हाट इट मींस. प्लांट वाले एसएसआरएनए, एनिमल वाले या तो सिंगल स्ट्रैंड आरएनए या तो डबल स्ट्रैंड आरएनए या तो डबल स्ट्रैंड डीएनए. मतलब यूजुअली एनिमल वायरसेस विल नॉट हैव सिंगल स्ट्रैंड डीएनए. ये नहीं होगा एनिमल वायरसेस में. ओके, एंड वायरसेस दैट इन्फेक्ट बैक्टीरिया कॉल्ड बैक्टीरियोफेज, दे हैव डबल स्ट्रैंड डीएनए. अगेन दिस इज रिटन इन द स्लाइड, जस्ट रीड फ्रॉम हियर. अब देखो वायरस में क्या होता है. वायरस हैज अ प्रोटीन कोट. ये तो कोट हुआ ना, इट हैज अ कोट ऑफ प्रोटीन्स एंड इट हैज सर्टेन एंजाइम्स आल्सो. दीज एंजाइम्स आर आल्सो प्रोटीन. तो इस वायरस में देयर इज अ कोट ऑफ प्रोटीन, देयर आर एंजाइम्स दैट आर प्रोटीन्स. जो कोट ऑफ प्रोटीन है ऑफ अ वायरस, दैट इज कॉल्ड कैप्सिड. कैप्सिड, दिस इज द कोट ऑफ प्रोटीन्स. एंड इसके छोटे छोटे यूनिट होते हैं कैप्सोमियर. मतलब कोट अगर मैं देखूंगा पूरा एक प्रोटीन नहीं है, ऐसे बहुत सारे प्रोटीन से बना हुआ है. तो ये जो वायरल कोट है, दिस इज कॉल्ड द कैप्सिड मेड अप ऑफ मेनी प्रोटीन्स. ओके, अब इसकी अलग शेप्स हो सकती हैं, हेलिकल हो सकता है, पॉलीहेड्रल हो सकता है, नॉट इंपॉर्टेंट. एंड वायरसेस कॉज मेनी डिजीजेस इन प्लांट एंड एनिमल. अब एक चीज में कंफ्यूजन हो सकती है एक टर्म में, वायरॉइड. वायरॉइड क्या होता है? इन 1971 देयर वाज अ साइंटिस्ट कॉल्ड टीओ डीनर. ही सॉ दैट देयर इज पॉसिबल दैट सिर्फ एक आरएनए मॉलिक्यूल बाय इटसेल्फ कैन एग्जिस्ट. So it did not even have a protein coat. It was just an RNA molecule. It was causing a disease in plant called potato spindle tumor disease. Potato spindle tumor disease कराने वाला सिर्फ एक RNA molecule था, आसपास कुछ नहीं था, क्योंकि उसमें कैप्सिड भी नहीं था, उसमें वो प्रोटीन कोट भी नहीं था. It was called a viroid. Okay, that's all you need to know. Okay. Now the questions with regard to whether viruses can replicate or not. I hope कई लोगों को तो ये क्लियर है, जिनको नहीं है we'll see after 2:30. इससे ज्यादा इंफॉर्मेशन फॉर वायरस इज नॉट नीडेड एक्सेप्ट द डिजीज कॉजिंग वायरसेस व्हिच वी विल सी इन डिटेल. जो ह्यूमंस के डिजीज कॉजिंग वायरसेस हैं, किसमें सिंगल स्ट्रैंड आरएनए है, किसमें डबल स्ट्रैंड डीएनए है, सब देखेंगे एट द एंड ऑफ इसी स्लाइड्स में. नाउ वायरल वेक्टर वैक्सीन वर्सेस डीएनए वैक्सीन. ये क्वेश्चन कई लोगों को था. वायरल वेक्टर वैक्सीन क्या है, नाम से समझो. वायरल वेक्टर वैक्सीन इज अ वैक्सीन दैट इज यूजिंग अ वायरस एज अ कैरियर. मैंने एक वायरस था, उस वायरस में एक जेनेटिक मटेरियल था, उस जेनेटिक मटेरियल में इंफॉर्मेशन थी टू कोड फॉर प्रोटीन्स एंड टू कोड फॉर एंजाइम्स। कुछ प्रोटीन कौन से थे जो उसका कोट बना रहे थे, जैसे कैप्सिड, एंड सम प्रोटीन्स वर एंजाइम्स। नाउ इट हैड द एबिलिटी टू कोड फॉर दीज एंजाइम्स एंड प्रोटीन ओनली, but it did not have ribosomes, right? It had the genes, but it did not have ribosomes. So what it was doing, it was using my ribosome or my cellular machinery to form this, to become what it was. Okay, what did I do in a viral vector vaccine? I edited its RNA and DNA. जो भी था उसके पास मैंने उसको edit किया. I made it a safe virus कि infect ना कर सके. I made it a non-replicating virus कि ज्यादा replicate ना हो, इतना replicate ना हो कि मेरा cell फट जाए, ठीक है. Then I modified it and I put the antigenic DNA in it. That became a viral vector vaccine. So I used a virus as a vector to deliver genetic material that was the genetic material of the antigen which I wanted to be expressed, right? Then there is DNA vaccine. Viral vector is clear. मैं वायरस को यूज करके डीएनए डिलीवर कर रहा हूं. डीएनए वैक्सीन अगर समझना है तो पहले एमआरएनए वैक्सीन याद करते हैं. एमआरएनए वैक्सीन में व्हाट वाज देयर, दिस वाज लिपिड और साथ में एक एमआरएनए का स्ट्रैंड था. तो वैक्सीन में जस्ट एमआरएनए था, कोई वायरस नहीं, कुछ नहीं. तो डीएनए वैक्सीन में भी यही है. डीएनए वैक्सीन इज जस्ट डीएनए, नथिंग एल्स. एंड व्हाट इज दैट डीएनए? प्लाज्मिड. डीएनए वैक्सीन में देयर इज नो वायरस, नो वेक्टर, ओनली द प्लाज्मिड. और प्लाज्मिड के अंदर मैंने इंटरेस्ट जीन वाला डीएनए डाल रखा है. दिस इज अ डीएनए वैक्सीन. प्लाज्मिड के अंदर अगर मैं एंटीजन का डीएनए डालूं एंड आई यूज इट, इट विल बी डीएनए वैक्सीन. इफ आई पुट द इंटरेस्ट डीएनए इनसाइड अ वायरस एंड देन यूज द वायरस टू गेट इट, इट विल बी वायरल वेक्टर वैक्सीन. दिस इज ऑल द डिफरेंस दैट यू नीड टू नो. Okay, there is DNA vaccine and recombinant vaccine. ये differences यहाँ लिखे हुए हैं अगर किसी को पढ़ने हो तो, right? But I think basically समझ आ चुका होगा. Recombinant अगर आपको पता है, DNA vaccine तो आपको बता ही दिया, simple. Plasmid के अंदर we are putting the gene of interest. तो ये molecule क्या है? It is just DNA, right? So it is a DNA vaccine. Recombinant vaccine can be viral vector, can be bacterial vector. इसमें क्या होगा, मैं वायरस और बैक्टीरिया को मॉडिफाई करूंगा एंड आई विल यूज द वायरस और बैक्टीरिया टू कैरी द रिकॉम्बिनेंट डीएनए मॉलिक्यूल. ये कल डिटेल में समझाया था. रिकॉम्बिनेंट वैक्सीन कैन आल्सो बी रिकॉम्बिनेंट प्रोटीन बेस्ड. इसमें मैं क्या करूंगा, दैट आई विल मेक अ प्रोटीन. आई विल मेक अ प्रोटीन ऑन द बेसिस ऑफ रिकॉम्बिनेशन. या तो मैं सेल में ही वो एंटीजेनिक प्रोटीन बनाऊं या फिर मैं बाहर बना के उसको दे दूं. बोथ दीज विल बी रिकॉम्बिनेंट वैक्सीन. ओके, and again more on this at the end of the class if you need it, but I think we have done it in enough detail और इतनी डिटेल में वो नहीं पूछने वाले. Okay, now let's come to nutrients and their roles. Okay, nutrients and their roles and the diseases that they cause. अब हम काफी सारी diseases पढ़ने वाले हैं. अब प्रॉब्लम क्या है कि आपको ये diseases रटनी पड़ेंगी. You will have to mug them up. There is no other option, क्योंकि हर साल एक या दो क्वेश्चन आ जाते हैं. And if I start explaining every disease, तो आप डॉक्टर बन जाओगे, right? So the problem is that you'll have to do some mugging up. So I will recommend that you be slightly attentive. Try to remember what you can here only, because there is no other solution to this. I can make it as simple as possible for you, but some mugging up will have to be done because there are a lot of questions on diseases. Okay, so now let's start with certain diseases. पहली डिजीज हम पढ़ रहे हैं क्वाशियोरकर. क्वाशियोरकर इज अ डिजीज दैट हैपेंस इन दोस चिल्ड्रन दैट आर प्रोटीन डेफिशिएंट. इफ यू डोंट गेट प्रॉपर प्रोटीन, you will be deficient in protein. Let's see here. There are three basic major nutrients that we have: carbohydrates, proteins, and fats. Carbohydrates provide energy. Proteins are building blocks of muscles. I hope प्रोटीन में कोई डाउट नहीं है. एक प्रोटीन है जो हमारा सेल बनाता है अमीनो एसिड करके, एक प्रोटीन है जो हम खाते हैं, खा के वो अमीनो एसिड्स में ब्रेक होता है. We have discussed this. This is the protein that we eat. The protein that we eat is the building block of muscles and building block of many other things. Like we have seen, it is part of many enzymes. जब हम बाहर से प्रोटीन खाएंगे तो हमें अमीनो एसिड मिल जाएगा, then our cell will use that amino acid. We have seen this before. So these are the three major nutrients or macronutrients: carbohydrates, proteins and fats. Fats are responsible for storage of energy, carbohydrates are responsible for generation of energy, and protein, as we have seen, are enzymes also important for muscles. Doubt regarding protein is clear. Now let's see major deficiency and issues. If you have protein deficiency, you will get kwashiorkor. Kwashiorkor maybe looks like this. Okay, as you can see, बहुत ज़्यादा the edema is happening. It's a swollen body, and he will not be attentive. His development will not be proper. All you need to remember for the prelims, kwashiorkor is protein deficiency. If you have both protein and energy deficiency, so you have both protein deficiency and carbohydrate deficiency, meaning your diet is so poor, neither is your diet giving you enough proteins, neither is it giving you enough carbohydrates, then you will have a disease called marasmus. A marasmus baby looks like this. It is shriveled, sunken, बिल्कुल ही growth भी नहीं हो रही है, right? It is stunted, wasted. Kwashiorkor baby looks like this. Swelling welling होगी, but protein is not there. यहाँ ना protein है ना energy है. अच्छा, ये तो हुए हमारे macronutrients. Fat deficiency just generally does not happen because we have enough of it, and वो कोई बहुत essential चीज भी नहीं है. Like carbohydrates and proteins are much more essential. But there are certain other nutrients called micronutrients. So far we have only studied macronutrients, the major nutrients. Micronutrients are small nutrients, certain other small nutrients. These certain other nutrients also have an important role to play in the body. These micronutrients are vitamins and minerals. Why are they called micronutrients and why are those called macronutrients? Macronutrients are required in large quantity. Lot of macronutrients are required. Micronutrients are required in much smaller quantity. You need much less vitamins and minerals than you need carbohydrates and fats, right? Macronutrients large quantity, micronutrients less quantity. Micronutrients में आते हैं vitamins and minerals. ठीक है? देखो, micronutrients are required in less quantity does not mean they are less important. They are also equally important. And if you have read in social issues of an issue called hidden hunger, okay, this hidden hunger is basically when people have deficiency of micronutrients, because after the green revolution we have a rice and wheat diet. तो उससे हमें कार्बोहाइड्रेट मिल जाते हैं, चलो एक तरफ प्रोटीन भी मिल जाते हैं, but we don't get access to vitamins and minerals because we don't eat vegetables and fruit. That leads to hidden hunger or micronutrient deficiency. Why is it hidden hunger? Because it's not immediately apparent, but the quantity required is less, but still it is needed, it is important. अब हम पढ़ेंगे कि विटामिन्स और मिनरल्स ना होने से कौन कौन सी डिजीज होती है. देयर इज नो ऑप्शन बट टू मग देम अप, सो आई विल जस्ट रन थ्रू देम क्विकली. देखो, विटामिन्स आर ऑफ टू टाइप्स. दे कैन बी फैट सॉल्युबल और वाटर सॉल्युबल. फैट सॉल्युबल और वाटर सॉल्युबल, क्या फर्क होता है दोनों में एंड व्हाई आर देयर टू टाइप्स, दैट यू डोंट नीड टू रिमेंबर. फैट सॉल्युबल में 4 ही विटामिन आते हैं, ए, डी, ई एंड के. विटामिन ए, विटामिन डी, विटामिन ई एंड के. Water soluble vitamins are of two types, vitamin B and vitamin C. Okay, what does water soluble mean? That you will be able to excrete them through the urine. Fat soluble will get stored in your fat and they will not be excreted through urine, right? This is the main difference. अब जो हमें इनकी मेन चीज पढ़नी है वो यही है कि what is the deficiency caused. If you have vitamin A, right, you will have night blindness. आप में अगर विटामिन ए नहीं है, विटामिन ए इज इंपॉर्टेंट फॉर फंक्शनिंग ऑफ द आईज, so you will not be able to see at night. Vitamin A causes night blindness. Vitamin D causes, like the deficiency of vitamin D causes rickets. Rickets are weak bones because vitamin D is important for the functioning of your bones. So if you don't have vitamin D, you will have bone problems known as rickets. Vitamin E is responsible for forming blood cells. So if you don't have vitamin E, you will have anemia. Anemia is of many types. Anemia can happen due to iron deficiency also. So vitamin E is also responsible for forming blood. So anemia means deficiency of blood. So vitamin E, if you don't have vitamin E, you will have deficiency of blood. Disease will be called anemia. Vitamin K. Vitamin K is important in clotting. So if you don't have vitamin K, you will have bleeding problems. 4 vitamins we have seen. A for night blindness, D is for rickets, E again responsible for blood formation, so you will have anemia, and K for clotting. Now vitamin B has many types. There is B1, B2, B3, B4, all of that, and there is vitamin C. तो पहले हम विटामिन सी पढ़ लेते हैं. These are water soluble. विटामिन सी से जो डिजीज होती है वो होती है स्कर्वी. In scurvy, the corners of your lips will start bleeding, your gums will start bleeding, because vitamin C is responsible for proper oral hygiene and developing like the mucosa in your mouth that is dependent on vitamin C. So if you don't have vitamin C, you will have a disease called scurvy. Now, these are the diseases for vitamin B. It's very less chance that they will be asked because ये काफी detailed हो जाएगा. But I have written them down, and again there is absolutely no option for you but to mug it up. So I am expecting कि आप ये slide से पढ़ के याद कर लोगे. जो important वाली है मैं बता देता हूँ. B1 causes a disease called beriberi. This one you have to remember. Then B9 is folic acid. Folic acid is a very important vitamin. B9 or folic acid is very important for the formation of your spinal cord. So if young children or mothers don't have B9, mothers don't have folic acid, तो उनके बच्चे के spinal cord की development proper नहीं होती. So that disease is called neural tube defect. Neural tube, nervous system की tube, spinal cord does not develop properly and they get folic acid deficiency leading to neural tube defects. Then there is vitamin B12 or cyanocobalamin. This causes megaloblastic anemia. It is a different type of anemia, right? Baki is not that important, but I will still expect you to at least read once. ये A, D, E, K, B1, B9, B12, C, ये तो आपको याद करना ही है. This you cannot skip. Okay, all right. Now let us read diseases caused by pathogens. पहले हमने क्या पढ़ा है? We have read diseases that are caused due to deficiency of certain nutrients. Second type of disease are those that are caused by pathogens. Pathogen, as you know, pathogen, disease generating. Okay. Now diseases are of these 5 types. There can be virus as pathogen, there can be bacteria, there can be protozoa, there can be fungi, right? Virus आपको पता है, bacteria आपको पता है, fungi आपको पता है, protozoa I'll explain. Okay, protozoa are nothing, they are single celled organism. जैसे protista थे, we had read something similar to that. So these are the groups of microbes that are causing diseases in humans. Virus, bacteria, protozoa, fungi. इन सबसे डिजीज होती है. अब इनसे कौन कौन सी डिजीज होती है वो हम देखेंगे, बट पहले अंडरस्टैंड व्हाट इज द रोल ऑफ अ वेक्टर. द रोल ऑफ अ वेक्टर, अ रोल ऑफ अ वेक्टर इज दैट ऑफ अ कैरियर. हमने वेक्टर डीएनए पढ़ा, रिकॉम्बिनेंट वेक्टर वैक्सीन पढ़ा. वेक्टर क्या है? वेक्टर कैरियर है. तो अगर कोई ऑर्गेनिज्म है जो इनमें से किसी का कैरियर है, वो खुद डिजीज नहीं कराता, but it is the carrier of the pathogen. The carrier of the pathogen will be called vector. For example, if a mosquito brings the protozoa causing malaria to my body, the mosquito is the carrier and the protozoa is the pathogen. So mosquito is the vector and protozoa is the disease causing organism. ये बात समझ आई कि disease causing organisms हैं, there is a vector, vector is carrier. Now, type of transmission. Transmission means spread of disease from one person to the next person. From person 1 to person 2 कैसे disease transmit होती है. Now this is very important. Okay, आप ये सीख जाओ कि transmission कैसे और इन पांचों patterns में क्या होती है, तो it will become much easier for you to remember diseases. देखो, first type of transmission is feco-oral. Okay. इसमें क्या है कि दिस इज अ पर्सन. ओरल मीन्स थ्रू द माउथ. So they have eaten a bacteria. They ate some bad food, drank some bad water. They got bacteria in their digestive system. अब उनके डाइजेस्टिव सिस्टम में तो उनके पेट में उनके इंटेस्टाइन्स में तो वो बैक्टीरिया इन्फेक्शन करेगा ही. बट व्हेन दे एक्सक्रीट फीसीज, this feces will also have that bacteria. This is clear? You ate bad food, having bad bacteria. Bacteria started multiplying in your body. When you excreted feces, when these feces came out of your body, this feces also had that disease causing bacteria. Okay. Now what will happen? Let's say a housefly or a normal fly. Okay. This fly comes and this sits on this feces. I know काफी gross हो रहा है, but this is how it actually happens. A fly comes and sits on these feces. तो इस fly के leg से bacteria attach हो गया. Bacteria is now attached to the leg of the fly. Now this fly goes and sits on somebody else's food. Person two's food, the fly sits on. Right? The fly sits here. The bacteria has now come here. Now this person will eat it and it will continue. This is feco-oral transmission. If a disease is happening in your intestine, okay, in which is happening that is giving you diarrhea, or it is giving you loose motions, it is giving you constipation, any intestinal problem, it has to be transmitted with this route. It is possible that children were playing and this they were playing with a ball and the ball fell inside the feces. They did not wash their hands before eating their next meal. So they got the bacteria from the feces into their body. It is possible that this feces went into a river because the sewage was not treated and then this water was drunk by people. So this bacteria then came into the body. So this is feco-oral transmission. यहाँ पर जो housefly कर रही है, housefly is a perfect example of vector. It is carrying the bacteria, but it is not itself causing the disease. तो जो भी disease intestine में हो रही होगी, diarrhea करा रही होगी, that will have this transmission. Okay. Second type of disease देखते हैं. Second type of disease transmission is droplet. Droplet transmission means this is a person. He has certain disease in his lungs. So when he will sneeze, when he will cough, certain parts of the body will release certain droplets. In droplets में भी वो bacteria होगा. हाँ पे ये bacteria है. So when they cough, when they release them, this bacteria will be there in the droplets. Now these droplets will be inhaled by some other person. तो ये भी लंग में चला गया. And this is droplet infection. Okay. तो जो भी diseases lungs में infection करा रही होंगी, खांसी करा रही होंगी, उनमें क्या example होगा? Droplet infection का example होगा. जैसे COVID-19 is classic example of droplet infection. Now third type of disease transmission is blood, serum or sexual transmission. Blood, serum or sexual. This means that the microbe or the pathogen is present in your body fluids. Body में कई fluids हैं. जैसे कि blood is a body fluid, saliva is a body fluid, sperms or semen is also body fluid. Okay. So in this case, the pathogen stays in your body's fluids. So if blood goes from one person to another, मान लो I use a needle and you use the same needle, and if my blood is infected, then from my blood the bacteria goes into your blood. This is blood-based transmission. Sexual or serum transmission, how does that happen? If two individuals have sexual intercourse, the sperm of the male will be going into the female, so there will be exchange of body fluids. So if the first individual is defective, even some part of the female's body fluid is going into the male. There is exchange of body fluids, right? Not going into details, but I hope you understand. So when in sexual intercourse there is exchange of body fluids, if one person is infected, the other person can get infected, right? So this is sexual transmission. Then third, इसी में आती है something called vertical transmission. What is vertical transmission? वर्टिकल मीन्स फ्रॉम मदर टू चाइल्ड। मान लो मदर हैज सम इन्फेक्शन इन हर ब्लड, बट जब चाइल्ड व्हेन शी इज प्रेग्नेंट विद द चाइल्ड, उनका ही मदर का ही ब्लड चाइल्ड में भी जाता है, मदर से ही चाइल्ड न्यूट्रिशन ले रहा है, मदर से ही चाइल्ड सारी एनर्जी और सारी चीजें डिराइव कर रहा है। वॉट एवर द मदर इज ईटिंग, द बेबी इज ईटिंग लेटर, राइट? वो मदर की बॉडी ही बेबी को न्यूट्रिशन प्रोवाइड कर रही है। सो इफ मदर्स ब्लड हैज अ सर्टेन एंटीजन, सॉरी नॉट एंटीजन, if a mother's blood has a certain pathogen, certain disease causing organism, then the child will also get a similar disease, right? So this is vertical transmission. This is similar to blood, क्योंकि blood में है या serum में है, that blood is going from mother to child. So sexual transmission we have seen, blood serum we have seen. Okay, now there is something called wound breaks. zoonotic पे आते हैं। wound breaks क्या होता है? like this is my skin and I fall down, तो ऐसे एक break हो गया skin में। so from this break a bacteria can enter। इसका हमने एग्जाम्पल भी देखा था टेटनस। दिस इज वूंड ब्रेक, मतलब स्किन में ब्रेक आएगा तब होगा। इट कैन ऑल्सो हैपन इन अ डिजीज कॉल्ड लेप्रेसी। लेप्रेसी में जो लोग होते हैं उनके हैंड्स पे सर्टेन लीजन्स हो जाते हैं। व्हेन यू टच अ पर्सन्स हैंड हु हैज हैड लेप्रेसी और यू शेयर अ टॉवल विद देम एंड योर स्किन ऑल्सो हैज सर्टेन ब्रेक, द बैक्टीरिया विल फर्स्ट कम फ्रॉम द hand of the infected person to the towel, then from towel into your skin. Okay, all right, this is then zoonotic. zoonotic means transfer from animal to human। कि ओरिजिनली ये डिजीज ह्यूमन में होती ह्यूमन में नहीं होती थी, इट हैपेंड इन एन एनिमल, बट ड्यू टू क्लोज कॉन्टैक्ट, ड्यू टू सम रीजंस, इट हैज कम फ्रॉम एनिमल टू ह्यूमन। ओके, दिस इज zoonotic infection. So these are the types of transmission, and in any disease you can guess the type of transmission after seeing what sort of impact it is having. Let's say I say HIV. HIV is there in the body's blood. HIV, human immunodeficiency virus causing AIDS, blood में immune system को खत्म कर रहा है, T cells को मार रहा है। So if it's in the blood, right, it will be going through blood transmission. And blood transmission means it is in the body fluids, so it can also go through sexual transmission. It can also go through vertical transmission. So if you have sexual intercourse with a person who has HIV, you will get HIV AIDS. If a mother who has HIV has a baby, the baby will get HIV AIDS. But if you share food with a person who has HIV AIDS, you will not get HIV AIDS. Why? Because fecal-oral transmission नहीं है। If you shake hands with them, you will not get AIDS, right? Because wound break से transmission नहीं है। This is why it is said that people who have HIV, don't discriminate against them, don't exclude them. You can share food with them, you can sit with them, you can talk to them, and they need it. They need sympathy, right? So, because the transmission only happens in this route, only this sort of contact will lead you to getting the infection, right? So, आपने types of transmission समझ लिए? Okay. Now it is already past 2:30, so I am giving you a little bit of homework and I'm stopping the class here. I'm stopping the class for those people who wish to leave. Your homework is that when you see these slides, you will see this list of diseases. ठीक है? इस disease list में क्या है? Disease का नाम लिखा है, इसका organism क्या है? पहले viral diseases हैं, फिर bacterial diseases हैं। इसका नाम क्या है disease का? इसका organism क्या है? इसमें क्या symptoms होते हैं? देखो, जैसे अब इन्फ्लुएंजा लिखा है, इन्फ्लुएंजा इज कॉज्ड बाय आरएनए वायरस, मतलब इस वायरस का जेनेटिक मटेरियल आरएनए होता होगा। इट इन्फेक्ट्स द रेस्पिरेटरी पैसेजेस, मतलब ब्रीथ जहां से हम करते हैं वहां से इन्फेक्ट करता होगा। तो व्हाट विल बी द मेथड ऑफ ट्रांसमिशन? इफ इन्फ्लुएंजा इज कॉजिंग रेस्पिरेटरी इन्फेक्शन, व्हाट विल बी द ट्रांसमिशन मेथड? ऑफ कोर्स इट विल बी ड्रॉपलेट, बिकॉज लंग्स में इन्फेक्शन है, तो जब मैं छींकूंगा या मैं खांसूंगा तो वहां से जाएगा, राइट? And is a vaccine available or not? So this table is there, right? I have taken this from a book. Okay, I would just request that you read this once. एक बड़ा सा टेबल है, आपको लग रहा होगा these are too many diseases. एक बार आप पढ़ो, एक बार मैं आपके साथ पढ़ता हूँ। Just read these diseases once. Okay, don't read these slides if you don't want to. ये COVID-19 म्यूटेशन ये सब मैं बता दूंगा, बट ये जो टेबल है, you read this so that next time tomorrow we can get through this quickly, right? यही है mainly health में we have to remember these diseases जो अदर मेन कंसेप्ट वैक्सीनेशन था वो हम पढ़ चुके हैं.